Hello, everyone, and welcome to week 7 of the 18th annual Symposium on the Conservation and Biology of Tortoises and Freshwater Turtles. Today's sessions are going to focus on field work and conservation actions. And first up, to start us off, Campbell to tell us about protecting freshwater turtle nests. Mariana, I'm going to kick it over to you.
Thank you, David. I will share my screen now, if that's all right. And here we are.
Mariana, I currently cannot hear your audio, but
can you not hear me? Is this okay? Can you guys hear me?
Yes.
Thank you, Jordan. Yeah, cool. Well, thank you for the introduction, David. It's really great to be back at the TSA. And today I'm here to talk about one of our manuscripts that has just been published in Biological Conservation. And I'm very excited because when I mean just published, I mean literally being available online 2 days ago. Hence the GOI there. I hope everyone can access and enjoy the read. And today, yes, I'm here to talk about assessing the efficacy of protecting turtle nests as a conservation strategy. This study has been a great collaboration between community group and academia. And a lot of the members here will recognize the person in the photograph in my slide. That's Marilyn Connell, who is member of the Landscape Group, also a former student at CDU, at Charles Darwin University, who has been working on protecting turtle nests since the year of 2000. So as we all know, protecting turtle nests is a common strategy used worldwide to protect freshwater and marine turtles. And however, we sometimes do have a big problem, which is assessing the effectiveness of conservation actions, such protecting nests, upon the population recovery. It can be a big challenge. And this is due to such a long time for turtles to achieve sexual maturity and generate offspring. So monitoring trends in population change can take up to 30 years, sometimes even more. So our study aimed to develop a methodology that could assess the efficacy of protecting turtle nests upon the population recovery in a much shorter period of time of a few years. Our case study, we selected the Mary River turtle, Elusoma crurus. I hope, and I'm sure some of you will recognize the photo. It's a freshwater species here in Australia, listed as endangered by the IUCN Red List, and also by the Australian Environmental Protection and Biodiversity Conservation Act. And for those not familiar with the Australian legislation, this is our piece of legislation that assists with our conservation actions. And a community-driven nest protection program has been protecting Elusoma crurus nests since the year of 2000. This study started in the year of 2015, so I'm going to focus upon the first 15 years of nest protection conducted by the community group. They protected 631 Elusoma crurus nests on those 15 years. And that meant approximately 9,465 hatchlings successfully emerging from their nests. And for our study, the Mary River posed a great scenario. We could select 2 different areas in the catchment to conduct our study. The Mary River turtle is endemic to one single river system here in Australia, and I have highlighted that in the map. So the river in blue and the shaded gray area is the catchment. We selected study area 1. It's a large part of the river And that is where the nest protection program has been occurring since the year of 2000. We also select a part of the river, which is our study area number 2, also highlighted in red in the map, is an area without such long-term nest protection in place. Our aim was to compare the population on those two stretches of the river. Those sites are approximately 65 kilometers apart, and they are also separated by a stream gauging weir. To do our study was vital to assess the population of each one of those study sites, and we chose to do a mark and recapture study. For that, we select a standardized passive capture method, fyke nets or funnel nets, known both ways. As you can see here in the photograph, they were set with the wings facing upstream, always set in the afternoon, and the trap was checked in the morning. And they were placed in the exact same locations every time. So we conducted 2 sampling events per year, one in spring and one in autumn, for 3 consecutive years, 2015 to 17. For each of those sampling events, we deployed 10 nets across each one of our study sites, and they were out for 4 nights. So every morning during those 4 nights, we would go and check the nets, release all the fish bycatch, and collect the turtles, measure sex, speed tag, photograph, and release them back in the river. The next step was to do an age estimation for Elusoma crurus. So we established a length at age relationship for Elusoma crurus using repeated records of age and length from non-aged individuals. And the result is this plot on the right-hand side, in which you can see the straight carapace length on the y-axis in millimeters, and their respective age in years on the x-axis on the bottom. We developed this curve in R using 3 different models. We don't have enough time here on the talk for me to go through it, so please read our paper. I hope you enjoy it. But basically, this is really important to see that I'm gonna highlight here why we did this. We did this because we wanted to see how big 15-year-old turtles are, or Mary River turtles are. So our curve indicated, if you follow the red dashed line on the plot, that a 15-year-old Elusoma crurus is approximately 317 millimeters in carapace length. The shadowed areas, both in gray and red, are the confidence intervals. This length at age curve was used to classify the individuals that were likely hatched prior to or during the period when the nest protection program was operating. This was vital for us to execute our population model. In this plot, we have our captures. So on the y-axis, we have the number of individuals, and on the x-axis, their respective straight carapace length. Colors indicate sex, with the dark green being the juveniles captured, the orange females, and the light yellow males. The dashed line that we have there on going vertically indicates the 317 millimeters carapace length. So turtles to the left of the plots were likely to be younger than 15 years old, and the ones to the right likely to be older than 15 years old. We run our models in R. We run a pop-on. We use the package R mark for those interested, but again, I won't give it too many details. And just remembering, our study area 1 was the one that had the nest protection program in place, and the study area 2 did not. And I guess the first thing we can all see at first is that we did, even though our method of capture was pretty standard across both the study sites, we did not capture as many younger than 15-year-old turtles in the study area 1. Our modeled population size for this area was 856 individuals, of which 16% were estimated to be below 15 years of age. This seems okay. However, when we compare to study area 2, we see that the story is a bit different. In this particular study area, without any nest protection program, things were a bit better. We had estimated 1,221 individuals, of which 33% were estimated to be younger than 15 years of age. This is our main conclusions. There is a lot more to it that is on our paper, and unfortunately, I just don't have the time to talk about everything here. But the takeaway message is that 15 years of Elusoma cruise nest protection had, unfortunately, not led to the population recovery, despite hundreds of hatchling turtles entering the river each year. Hence, predation of eggs from the nests may not be the singular factor hampering population recovery, and therefore nest protection alone is not a sufficient strategy to reverse ongoing decline. The nest protection program has been vital to protect those eggs from terrestrial predators. However, our study indicates there is a lot more happening in the river once those hatchlings go in the water that we are still not aware of. And hopefully we'll have some more answers in the future with a project that is starting next year. This study also demonstrated how vital is to monitor the long-term impacts of conservation actions on the recovery of the populations. And lastly, the methodology developed in this study provides a technique to assess population status within a much shorter time frame than the traditional survey count methods. And I would like to thank all our partners, all our funding bodies, also those who provided data for our length at age curve. Special thank to all our volunteers, without whom we couldn't have done this. And here's a little snapshot of our just very, very recently published paper. That's the DOI in case someone wants to check it out tonight or today in America. Sorry, here is still half midnight. And for those interested on the Mara River Turtle Nest Protection Project, please check the community group, Tidal and Strict Land Care Group Facebook page and website that I have put it in here. And if you want to know what our lab is up to and what our research is on, here I have also provided our lab Facebook and website. And this is it for me today. Thank you everyone for listening. And if you have any questions, I am here to answer.
We will wait just a minute to see if we have any come over from YouTube.
Lovely.
And there is one on the way. Can you see it in the chat?
I can. Great. So I'll read it out loud because I'm not entirely sure who can see and who can't. So question 1, very interesting. Can you give a quick summary of the different growth models you tested? Yes, I can. I am gonna have to open my paper because I cannot remember their names just by the top of my head. But we use 3 different models. We tested, sorry, I need to pick them up because I can't remember. So we tested von Bertalanffy, Gompertz, and logistics. So we did the 3 models, and then we used obviously the AICC. There were less than 2, selected the best fitting model to be the one. And we also, on the paper, for those interested on the age at length curve, on the supplementary material, we executed the same for all the freshwater turtles as a comparison, and it is available on the supplementary materials on the paper as well. I hope I have answered your question. And then I have, are you changing sampling methods to catch more juveniles? No, the methodology was exactly the same for every study site, for every trap, for every event. Exactly, so we wouldn't be biased the capture towards any particular size class or sex. again, I hope I've answered. Question 3, great research, Mariana. Thank you, Jordan. Or if it wasn't Jordan, whoever asked that, because Jordan is just posting it. Sorry, yes. So thank you. What do you believe is driving the lack of recruitment? So we discussed a few options in the paper. One thing that was obvious in the data is that there is less females in that stretch of the river, and therefore less nests. So it's great that the community group is protecting the ones that are laid every year, because it was about twice as many females on the other study site. We believe there is another culprit in the river based on our bycatch, the fish bycatch. And this is a further study that we're going to start. There's a follow-up study that we're going to start next year. But the fork-tail catfish, it's a big problem in that particular section of the river, just downstream from study site 1, is an impoundment region. And we believe that that is favoring an oversized catfish population. Just so you have an idea, I actually, if I can, I'm gonna show you guys another slide that I put it in here, just in case. This is the fork-tail catfish, and as our bycatch on the study area 1 with the nest protection program, we collected 325 of those, and they are known for feeding upon hatchling turtles, which we only caught 20 on the other study site. So we are going to investigate if they are the main cause of the lack of recruitment in that area. So I hope I have answered the question. But in that, we are not entirely sure. We think that that's what's going on. This photo was taken in the river, is one of our catfish that had another fish inside its mouth, but this is actually quite a normal size catfish. I don't have a photo just now, but we collected some gigantic catfish on that stretch of the river, and we believe that the impoundment and the conditions there are favoring these oversized fish. I hope I have answered the question. So I'll go back into the other slide. Thank you.
Hi there. I am part of the Desert Tortoise Recovery Office for the U.S. Fish and Wildlife Service. And I'm going to be talking about our experience translocating captive tortoises of the Mojave Desert tortoise within southern Nevada. I'm going to be talking about survivorship. And so the project is larger than just our office and my colleagues there. The Great Basin Institute did the surveys to recapture marked animals, and the San Diego Zoo was involved in raising these tortoises and also setting standards, health standards before they were released. The Mojave Desert tortoise is classified as critically endangered by the IUCN.
Linda.
In states, it has protected status under the Endangered Species Act.
Linda.
And the latest one also describes a role for population augmentation.
Linda, we can't see your presentation. If you want to hit that share.
Excuse me?
We can't see your slides. If you want to go ahead and hit your screen share.
Is it visible now?
Yes, looks great.
But you could hear me before.
Yes, absolutely.
Okay, well, I'll just start here then. So it may be surprising that the Mojave Desert tortoises are kept as pets in the southwestern United States. Just a moment here. Here's my problem. Oh. Although they're protected here since 1990, and there's no collection from the wild, people who had pets before that time are able to keep them as pets, and their captive progeny are also legally kept as pets. So this holding facility in Nevada was set up in 1990 when the tortoise was given protection, approximately the same time. And this is the source of our captive tortoises, although the center was actually set up to be a halfway spot between wild to wild translocation. So tortoises removed from the path of development would be kept in this facility and then moved into other wild locations. However, the public hotline resulted not only in calls about tortoises that were in the way of danger, also people with pets were calling to have them picked up. They didn't want the pets anymore. And this may not sound like a large source of pets, but the largest city in the Mojave Desert is Las Vegas. And so you can imagine with a population like this, there are a lot of pets. And this shows where animals were picked up as a result of the hotline all over the Las Vegas Valley. And this number coming in yearly was quite high. When the San Diego Zoo started operating the center, the hotline was gradually shut down. And so you can see that fewer and fewer tortoises were being brought in. In 2014, the center lost funding and it was closed. So we were left with many tortoises, most of them captives, that needed to be relocated. So our question for the recovery office that I work in was, could we use these animals to recover wild populations? I mentioned that our recovery plan does include population augmentation as a strategy. And there had been successful short-term translocation, at least. There's a lot of experience moving wild animals out of the path of development to other areas. And in the short term, at least, their survivorship has been pretty high and solid. One of the issues with captive tortoises is that there had been previous translocations involving captives, but it was obvious that there were a lot of shell remains at those sites. And although there was not official monitoring, there was concern that captive animals might not fare as well. We proceeded anyway. We had the San Diego Zoo working with us to screen animals before they were released. And this is the site we chose. The line across the bottom of this map is a highway. 7 years before we released animals there, the highway was outfitted with a barrier fence so that tortoises couldn't get onto the fence. We know that animals that are released move extensively right after they're translocated, and even for a couple of years, they move as far as about 6.5 kilometers. So this is the area we set up for release north of the highway. We didn't release all the way to the end of the fencing, which is that red line. Instead, we kept the animals as far away from the edges as possible, 6.5 kilometers away. The upper line on this release area is an elevation line. Although tortoises do like the rolling hills in the desert, they don't generally go much above 1,250 meters. So that was how we outlined our release area. We released them over 2 different years. And then we followed up with surveys for 3 years. The elevation line forms one boundary of our survey area. The highway forms another. And then in the west, there's just an administrative boundary. But that area is probably outside the effect of where the captive tortoises would have moved to. The surveys to recapture them and to mark residents were conducted, not strictly linear surveys, but these are the paths that surveyors took. They would wander off to look in promising sites, cover sites under vegetation or in washes. So I'll start showing you some of our data. We released over 700 animals. I'm only talking about the ones that are at least 100 millimeters because they could hold a mark. We consider animals up to 179 millimeters to be juveniles, and animals over 180 millimeters can be reproductive, so I'll refer to them as adults. Little less than half of the animals we released were adults, which is the focus of most of what our recovery work is. The reciting rate for adults was adequate. It was quite low for the juveniles. So we ended up also using recovery of dead marked animals for the survivorship analysis. We found about half to 2/3 as many resident adults as we translocated, and also had a similar recapture rate. It's really hard to find juveniles, and so we don't have as many of those in the analysis. So I used program MARC, I used RMARC to evaluate the best way to describe survivorship. I compared translocated and resident status, also the two different size classes, juvenile versus adult. I looked to see whether there was a cost of translocation during the first year. They were translocated over 2 years, but in either case, I looked to see whether they had lower survivorship that year. This has also been done in other studies, and in tortoises, it was done by Bertolero et al. Also. I used constant survivorship as my null model. And the results are shown here for the different models I ran. They're rated based on their AIC values. And you can see that 3 or 4 of them have pretty good support from our data. So I'll just talk briefly about that top one, which is an interaction model of the translocation versus resident status, the size, and included that 1-year cost of translocation. The second model is an additive one. So it's similar to the first one, but no interactions. And then interestingly, the third best model only included that 1st-year cost of translocation. So I used a weighted average to get estimates of survivorship, and I'll explain, I hope this will explain why those top 3 and 4 models kind of all had a good amount of weight. So this is showing just the translocated animals just in their first year. And you can see that the survivorship of the juveniles is quite high, and the adults is also pretty high. The confidence intervals are a little bit wide, but still, it's high survivorship. And then after that first year, the translocated animals all had extremely high survivorship, and the adults really picked it up following their first year after translocation. The residents had high survivorship the whole time. And so this explains why the first year cost of translocation really was highlighted in the supported models. So conclusion just for this part is that captive tortoises have high annual survivorship, at least for the first 5 years. And this includes the juvenile tortoises. There was a 1st-year cost of translocation that was particularly noticeable in the adults that were moved. But after a year, translocated and resident tortoises had similar survivorship. I will just mention again that we're aware there are other studies that did not include monitoring, that it didn't look like survivorship went quite as well. So I'm going to talk a little bit about how vital rates are consistent or not with population success, because our goal isn't really to have individual tortoises survive, but we want this area of the desert to be filled in with adults. Comparing this to the classic paper in 1993 on Blanding's turtles, we had similar very high adult survivorship, and Congdon et al. Indicated that you would need fairly high survivorship in juveniles also, because to get at least 2% of them from hatching to adult in order to have stable populations. And we had this level of survivorship, but we didn't look at the smaller animals. Fortunately, there is another research group looking at that size class. They also, it looks like they're going to also show about over 82% annual survivorship for animals under 100 millimeters. However, the same group has noticed a concerning issue with the non-native invasive grasses that we have throughout the Mojave, and we have in large amounts at this translocation site. Those grasses have very poor nutritional quality and have been shown to decrease growth and increase mortality. in juveniles especially. So we'll be watching that in the future. And this gets at the fact that we really anticipate this being a long-term monitoring project with different metrics over time as we get to the generation time for desert tortoises, which is about 25 or 30 years, just to show that this was a successful translocation. And that is the end of what I have.
We're going to hang on just a minute to see if we have any questions come over from YouTube. Thank you, Linda. That was great. All right, and we don't have any questions. So thank you again, Linda.
Okay, thank you.
Okay, I think I'm up. My name is Ethan Kessler. I'm a postdoc at the Illinois Natural History Survey. And I'll be talking about some of my dissertation work here on the alligator snapping turtle and a reintroduction in Illinois. So we're dealing with this global biodiversity crisis right now, which is commonly framed with species extinctions, which are incredibly important. But across all animals, we're seeing incredible population declines as well, especially in freshwater populations, where we've seen an 83% decline in global freshwater populations in the last 50 years, which is incredibly devastating. Turtles are especially hard hit in our current human-dominated world. Over half of all turtle species are considered threatened, which makes it the most threatened of all the vertebrate groups. And turtles are especially hard hit because naturally they have a really high dependence upon high adult survival to offset naturally high juvenile survival rates or mortality rates. So with some populations naturally having almost 100% yearly adult survival. But when you mix in, human-derived effects on populations such as overharvest or road mortality or indirect effects such as habitat destruction, you end up with kind of a toxic mix for turtles where we lower those high adult survival rates and populations can crash really quick. And so the common question, at least that I get when I'm in the field, is who cares? And I think I'm in friendly company here. I'm kind of preaching to the choir. Freshwater turtles are incredibly important. All turtles are incredibly important. One reason that we can point to is that historically freshwater turtles had the highest recorded biomass per area of all vertebrates. And this can have incredible effects on ecosystems where turtles are shaping the trophic structure of these habitats. And when you have semi-aquatic turtles or nesting turtles, there's a huge transfer of nutrients from the aquatic to terrestrial habitats, which can have landscape level effects rather than just freshwater effects. And so increasingly we're using reintroduction in turtles, and reintroduction has gotten a bad rap and not for bad reason. I mean, there have been some less successful reintroduction projects, but with that being said, why do we choose reintroduction? And I think the most obvious reason is that it's a last resort in some cases, where if you have a population that's been extirpated or no longer exists at an area where they used to exist, how else are you going to conserve that population if the population doesn't exist? You have to reintroduce them back into the habitat. The Galapagos tortoise is an example of this, where some islands had the tortoise species had tortoises that were extirpated. They reintroduced them, and they've seen success thus far. But reinforcing declining populations is another important reason to do reintroductions and translocations of turtles. You know, we commonly see this in North America, where I'm from. Nest predators such as raccoons and other mesocarnivores are subsidized by human life. The abundance of trash and dog food and a number of other food sources that are available to these mesopredators really balloons their populations. And these are incredibly good predators of turtle nests. And so we see some turtle populations, especially in urban and suburban environments, where you have almost no nest survival. And turtles are equipped to handle this across, you know, a year or two. But if this is sustained over years and years and decades, then it leads to population extinctions. Where we can see in this kind of figure here, naturally we have hatchlings coming into the population every year. If that stops, then we only get older and older individuals in the population until all of those individuals die and the population is gone. So it looks like a population is lingering on and doing fine, and then one day it's just gone because all the adults have finally died off. And so we can reintroduce young or younger turtles into the population to keep that population structure and try to keep a healthy population structure until we can resolve conservation issues without the population going extinct in the meantime. So the study animal here is the alligator snapping turtle. It's the largest freshwater turtle in North America. I'm sure many of you are familiar with it. It is native to Gulf of Mexico drainages in the United States and is currently under consideration for a listing under the Endangered Species Act to receive federal protection from the United States government. In Illinois, there have only been 17 records since the 1880s, and only 4 of those have come in the last 50 or 60 years. And the last 3 came from the same tributary of the Mississippi River down in extreme southern Illinois. So this has kind of been identified as the last area for this species in the state. This is the site that we chose for releases. Every year we would release turtles in July or August and fit a subset of those with radio transmitters that we could, that allow us to track them and monitor them through time. We've released over 500 turtles since this project began back in 2014. And every year we try to manually recapture all of our turtles to monitor their survival, health, and growth. And we do this at the beginning and end of the active season in spring and fall. And recently we've run into a couple of roadblocks. We had extreme flooding in spring 2019, and then COVID-19 shut things down this year in the spring. But we've gotten gotten to be able to get out into the field and capture these guys pretty reliably since the start of the project. And so we use the data collected from this field work to develop population projection models. And traditionally, this has been done with stage-based matrix population models, which are incredibly powerful methods that allow us to estimate stage-specific estimates of survival, fecundity or reproduction, and transition probabilities. So each column here represents an age. Say this is hatchlings, juveniles, and adults. And we have our survival estimates here, our reproductive or fecundity estimates here, and then the probability that an animal in stage 1 is transitioning to stage 2 each year. And so we can use just these few estimates to determine how our population is going to function through time. Recently, there's been the development of integral projection models, which allow us to do the same thing, except do it continuously across all sizes. And this gives us, a finer scale estimate of our population. So we conducted a growth analysis. We had several nonlinear estimates or nonlinear models of growth that we tested, and then selected the one that best fit with AIC. And we found that the von Bertalanffy growth model was our best fit. We can see the growth curve here on the left, and then the estimate of fit here on the right. And these, this line here points to the age at sexual maturity of our animals, our estimated age at sexual maturity. This is the size at sexual maturity. We see that our animals aren't becoming sexually mature until age 26. And this is concerning because in the literature, the estimated age at sexual maturity is between 13 and 16 or 17 years old. So this might have devastating effects on our population. For our survival analysis, we use known fate model to estimate survival. And this is a popular model to choose with radiotelemetry. And then we tested several models to determine which factors influence survival. I'm not going to show all of them, but I'll show the best model here. Our results were complicated by a bycatch event, which happened in fall 2019, where 6 adults were killed in an abandoned illegal hoop trap that a local landowner had put out to catch fish. So we lost 6 adults from our population, where we had almost 100% survival of these age classes up until this point. So we modeled survival both with and without that bycatch event. We found that age and season were our best predictors for survival. We see decreased survival in the winter months. This is pretty common in turtles. And then we also see our size-based effect of survival here as well. We can see how the curve kind of changes with bycatch and then without bycatch in our population. And it does have a pretty extreme effect. While not extreme to the eye, the reduction of just a few percentage points in adult survival from year to year can have devastating effects in a turtle population. So for our integral projection model, we used our modeled growth as well as a modified growth model that made our modeled animals reach sexual maturity at age 16, which is what's reported in the wild. And then we use survival estimates both with and without bycatch. For fecundity estimates, we used estimates from the literature because we don't have this data for this population. And what we see, I'll walk you through all these scenarios. These are bootstrap estimates of lambda. Lambda is the an estimate of population growth, where all values above 1 indicate growing population and all values below 1 indicate a declining population. For our base model with all of our observed data, we see that there are no instances where we reach a growing population. In fact, we get pretty dire towards the left end of that curve. When we increase this the age at sexual maturity with our growth, we see that the outlook gets a little bit better. We have some instances where we potentially have a positive growth rate. When we remove bycatch and keep our observed growth, we see less variability in our estimates. However, it's still all negative population growth. And then when we remove bycatch and increase growth, we see that we are still centered around a declining population. However, we do have a lot more instances where we do have potential population growth. And so what we have is a situation where our bycatch isn't necessarily improving the picture too much, but it's leading to less uncertainty in our outcomes. But it's the growth that's really affecting our population. And when we model this out 25 years, we find that the totally altered scenario is the only one that results in a growing population. All other population estimates declined. So, our conclusions here is that survival is best explained by size and season. This is incredibly common in turtles, and maybe a little more exaggerated in our study because winter months are comparatively harsh in southern Illinois when compared to the rest of the range of the species. We find that preventing bycatch reduces the variability in our population growth, but on its own cannot lead to a stable population. And that our reintroduced population is declining, and it's largely due to slow growth rates, where we have individuals reaching sexual maturity almost 10 years later than wild populations. And this might be due to the fact that we're at the northern extent of their range, and maybe there's just not enough active season days for them to grow. So what do we do about this? For the survival, we can try to actively prevent illegal hoop trapping just to give our turtles the best chance at succeeding. But for growth, it's kind of a sticky issue because how do you improve growth? One thing that we can potentially do is try to improve habitat for juveniles and subadults. That would give them better forage or better thermoregulation, and hope that improves their growth. But it's kind of a tough issue, and so that's kind of where we're at now. With that, I'd like to thank everyone involved. This has been a huge project. There's a lot of partners, and I can take any questions that people might have.
We will give it just a minute to see if we get any over on YouTube.
Okay. How long was the stretch of stream you were working in, and how many turtles per mile would be needed for a healthy population? That's a good question. The streams that I was working in is a relatively small stream. I think from the most northern point that we recorded a turtle, and the most southern point was probably 6 or 7 miles. So not a huge length of river. The turtles per mile is a really interesting question. And I think that it depends on the size of the water body, because you just need to get a certain number of turtles to have a viable population. That's not something that we've directly assessed at this point. We're just trying to look at positive population growth. But for our population right now, I mean, we have, dozens of turtles per mile. Granted that most of them are juvenile turtles that we are releasing. So our second question is, historically, were Illinois populations self-supporting, or were they always augmented by influx? And this is the big question in Illinois. We have, in the last 150 years, we have 17 records, and there's just so little that we can learn from just those records, especially at a time, where most of these records were just oddities, and there's no real rigorous work being done with these turtles. There's people that think that turtles in Illinois came from southern states, and just old turtles that are able to go up the Mississippi River and stop over in Illinois and find them there. However, there have been smaller turtles that were found. The boot heel of Missouri is the closest region that has naturally reproducing populations. There's no evidence that alligator snapping turtles have ever successfully nested in Illinois. So probably there, you know, we don't know for sure, but there's no evidence of it. So. How much algae do you see on Illinois alligator snapping turtles? And does this differ from other species in the same habitat? We don't see a lot of algae on the turtles in our site. It's a pretty silty stream. There is a lot of canopy cover at our site. The turtles that we get don't really have a lot of algae on them. So we don't really see a difference because there's really no algae on any of the turtles that we capture at the site. Great, thanks a lot, everybody.
I'll share my screen. Okay, my name's Helen Truscott, and today I'm presenting work I did for my master's degree, looking at the effectiveness of conservation programs on the harvest of wildlife in developing tropical regions. And doing this by looking at the case study of the pig-nosed turtle in the Gulf region of Papua New Guinea and measuring the effectiveness of the PICO project after working on 10 years of conservation in the region. There we go. So the endangered pig-nosed turtle is found only in southern New Guinea and northern Australia. And in the study area in Papua New Guinea, there's been substantial decline in the last 30 years, and it's attributed to overharvest of the species. Most people in the region live traditional lifestyles, and they're still reliant on wildlife as a protein source. The area has really limited transport routes, and it's quite isolated. So this results in the pig-nosed turtle consumption and trade generally being locally confined. However, the region, like many developing tropical areas, is experiencing accelerated rates of economic and social change. And in 2010, there was a major LNG project which has increased connectivity to neighbours. This could potentially lead to a shift from a subsistence to a cash economy, putting further pressure on the species and making the PICO project's conservation efforts even more vital. So the PICO project is a project established in 2008. It's a community-led conservation program and aimed to achieve sustainable harvest for the pig-nosed turtle, as well as sort of support broader environmental messages. The project was jointly led by the University of Canberra, PNG Biological Research Institute, the University of PNG, and had Oil Search and ExxonMobil as major sponsors over the years. And now it's run through a recently formed not-for-profit, the PICO Biodiversity Network. So to date, as you can see up there on the screen, the project had a range of activities, research, working with schools, radio, consumption monitoring, surveys, protected nesting sites, as well as sort of broader conservation work as well. I think one of the issues is there's really a lack of reliable data on the success of community conservation programs. And there's an estimated 50% failure rate of environmental awareness campaigns actually resulting in the wanted behavioural change. So with the PICO project, prior to its project and its early partner projects, the area in PNG had a pretty limited exposure to modern conservation agendas. And to add to this, data was collected either side of a major LNG development. So therefore, the evaluation of the PICO project provides an opportunity in which to understand the effectiveness in conservation programs and changing not only attitudes, but behaviours. And I think the hope is by understanding the project's efficiency, it can guide future action in the preservation of the iconic species, as well as be adapted for use in similar culturally and environmentally complex regions. In 2009 and 2016, the PICO project undertook household surveys to evaluate the impact of its outreach activities. Surveys occurred over 7 villages, each representing different environmental, cultural, and social diversity in the area, and all with differing involvement with the PICO project. Collected information on harvest, awareness, attitudes, socio-economic indicators, among many other things. And with further information on trade and connectivity I gathered during the field. So I think, first of all, looked at changes in relationships to social economic factors to understand the background that the PICO project was working in. So from 2009 to 2016, there was no significant difference in households with a member in paid employment or the highest level of education. And we actually found that there was less households with access to working outboard motors. So from a very basic overview, it looks like the predicted improvement in social economic conditions in the area didn't occur, but obviously that would take some more examination. What did find was there was a significant increase in market price per pig-nosed turtle from 2009 to 2016, but there was no significant change in the mean egg price. When we looked at the number of turtles and nests harvested per household in 2016, we didn't find any relationship to harvest with any of those social economic factors tested. Next, looked at the awareness of the issue of pig-nosed turtle decline. Positively, there was a significant increase in awareness of decline in 2016 compared to 2019, with a 19% increase in surveyed households perceiving that the population is declining, which is reflective of the actual situation. But unfortunately, there was no significant difference in the number of pig-nosed turtles or their nests harvested between the two survey points, 2009 and 2016. Then I looked at awareness and harvest behaviours and found no relationship in 2016 between awareness of pig-nosed turtle decline and whether households harvested turtles or eggs. Separating out those households aware of population decline, we found that 76% attributed that decline to human activities and 66% directly with overharvest. So I guess this indicates that it's not a lack of understanding of causation that's resulting in awareness of population decline not being related to harvest behaviours. So next we looked for how the PICO project was related to awareness of pig-nosed turtle decline. And we found that household awareness was significantly higher in those villages with higher exposure to the conservation project. However, the awareness did not translate into action, and there was no significant difference found between number of households harvesting pig-nosed turtles between villages with high or low exposure to the conservation program. So I guess to sum up with the PICO project, it was very successful in increasing awareness of the issues facing the endangered pig-nosed turtle and its declining populations. Additionally, locals involved in the project became project champions, advocating for environmental messages. And I think the increased awareness of and the finding that the decline was overwhelmingly believed to be human-related is really positive in addressing the issues. Additionally, the project has increased the capacity of local people and expanded knowledge on the species. But despite these positive findings, it has not yet led to any detectable change in the harvest behaviour. So I think often with environmental studies, we kind of rely on measures of awareness of the issue to indicate a program success. And while measures of awareness can often be easier to ascertain than ecological outcomes, awareness is not synonymous with desired behaviour. And I think this study highlights the danger in assuming that awareness will automatically lead to action. And if we ignore that gap, we risk not achieving our conservation goals with pretty disastrous effect to our vulnerable turtle populations. So we looked at, why does awareness not lead to action? Because looking at the survey responses, local people in the area really want the pig-nosed turtle to thrive. But behaviour is, complex, and it's influenced by a much broader set of motivators than just awareness of the conservation message. I think especially in the developing tropical nations, economic, social, political, and cultural systems are really inseparable from conservation behaviours. And there's much more entrenched competing drivers that must be overcome in changing behaviour versus awareness, which I try to demonstrate in the diagram that you're seeing now. To add to these challenges, the PICO project is likely to face more competing drivers. In 2009, when I did the study, trade remained pretty localised, but there's a lot of future development planned for the region, and this is going to probably add to pressure and by opening new markets for pig-nosed turtle within the Southern Gulf. So I think, as I was saying, that the findings of the study kind of highlight some of the challenges for conservation of the species, but we can also use it as a framework for future efforts. I think the three main things we found, or I found, was for a program to be successful, it needs to be truly community developed and led. If conservation actions are adopted by a community rather than developed by a community, they're just not sustainable. There's a lot of underlying differences in community organisation, and these differences won't support externally initiated conservation once that external influence is removed. The next important thing for these programs is to acknowledge and support cultural differences and motivation and conservation ethic. I think many Western conservation principles ignore the intrinsic integration of cultural and biological systems and their importance to local people. So conservation programs working in this environment, they have to align and give legitimacy to different cultural frameworks, and they really have to work within these rather than alongside these. And lastly, programs have to address the social economic issues. They've got to recognise disparity in living conditions and people's desire for a better life. Projects really need to focus on local priorities and build capacity to enable communities to generate their own benefits rather than using a more sort of, one-off fee-for-service type arrangement. So back to the PICO project, we can say that the PICO project is aligned to these factors that we've identified. It's community driven, recognises local priorities and works within local cultural systems. And its approach has really built local capacity and created a new generation of locals aware of environmental issues and prepared for action. So I think by understanding and recognising the barriers identified between awareness and action and working to overcome these, the PICO project will continue its work on saving the pig-nosed turtle and work on that gap to turn awareness into action. I wanted to thank everyone involved in helping me put together that research, and I'll see if anyone's got any questions.
We will give it just a little bit longer to see if we have any questions come in. Thank you, Helen. That was fantastic stuff.
No worries.
While we're waiting here, up next is going to be Louise Streeting telling us about the effectiveness of nest protection as a conservation strategy for the endangered Australian freshwater turtle, Myocheles bellii.
Right. All right. Thanks, everybody.
Thank you, Helen.
Hi, everybody. My name's Lou Streeting, and I'm a PhD student at the University of New England in Australia. And my research is looking at the conservation and reproductive ecology of the endangered Bell's turtle, Myocheles bellii. My co-author, Martin Dillon, is an ecologist and manages the Turtles Forever conservation program for Bell's turtles. It's widely known that turtles are vulnerable to predation during early life stages. So eggs in the nest, hatchlings and small juveniles. Predation is particularly bad when predators are invasive species that the turtle hasn't co-evolved with. In Australia, predation of turtle nests by the introduced European red fox is decimating some freshwater turtle populations. And in the case of the Bell's turtle, current fox control is inadequate, and until we have effective fox control measures in place, conservation is required to ensure the persistence of this turtle species. One of the aims of this project is to quantify the extent of nest predation occurring for this turtle species. Bell's turtles are a short-necked pleurodirian or side-necked turtle with a restricted habitat range. They're a long-lived species. We know they live for at least 40 years, but we suspect that they live a lot longer. This is a large female. She has a carapace length of about 30 centimeters or 12 inches, and she weighs 3 kilograms or 6.5 pounds. Bell's turtles are a late maturing species, and they lay a single clutch of up to 30 eggs in the riverbank, and those eggs have to incubate in the riverbank for 80 to 90 days. Whilst we still have a reasonable number of adult Bell's turtles in our waterways, juvenile recruitment is thought to have all but ceased, and juveniles make up less than 1% of the Bell's turtle population. And so the population is aging, and without sufficient juvenile recruitment, this species is likely to eventually become extinct. So over 3 breeding seasons, we've found that more than 95% of Bell's turtle nests are raided by the invasive European red fox. You can see a fox on the left of the screen sniffing around a Bell's turtle nesting bank. And on the right, you can see a raided Bell's turtle nest. You can see eggshells scattered around the surface, and there's actually a fox scat in the nest chamber. This time-lapse footage shows a fox raiding 3 Bell's turtle nests on the one night. There are trail cameras mounted on this bank, and they're monitoring protected nests. However, the fox has come in and raided the unprotected nests. Unfortunately, we've also found that foxes are taking the adult females when the female comes up onto the riverbank to nest. This image shows some Bell's turtle carcasses scattered around an active fox den. This footage shows female Bell's turtles coming up onto the riverbank to nest during a rainy afternoon in spring. And at one stage, there's 4 females nesting simultaneously, and you can also see other turtles in the river scoping out the bank. This still photo just highlights those 4 nesting females. So we're looking at the effectiveness of nest protection to keep foxes out. We're placing wire mesh or steel cages over the top of live nests, and that protection stays in place for the next 80 to 90 days. Finding the turtle nests can be quite difficult. Sometimes you can see tracks leading from the water up the riverbank, and sometimes you can see a turtle-shaped imprint in the substrate, which indicates that a female turtle has nested. However, these tracks and traces are not always present, and when they are present, they're not present for very long. So we're using 3 strategies to find and protect Bell's turtle nests. So we're walking the riverbank on foot, searching for nests. We're setting trail cameras or camera traps on likely nesting banks. And we're also working with a detection dog team from Canines for Wildlife, who have trained 2 dogs to specifically find Bell's turtle nests. This is the typical habitat that Bell's turtles like to nest on. It's exposed and bare. There's very little overhead shade or vegetation, and it's quite often on a livestock access way. The soil type varies from sandy loam, rocky gravel, all the way through to hard setting clay soil. Here's another nesting bank. You can see that there's trail cameras set up here monitoring protected nests. Last season, we protected 14 Bell's turtle nests on this particular bank. The photo was taken late in the season when the hatchling turtles were emerging from the nests. So we have erected a drift fence and a pitfall trap to intercept the hatchlings. Over the 3 breeding seasons, all of the Bell's turtle nests have been found within 6 meters of the water, an average of 2.5 meters from the water, and an average of 1.5 meters above water height. No Bell's turtle nests were found on top of the bank in the pasture. And the turtles tend to nest more frequently at the base of a step in the bank. This footage shows a fox trying to get into one of our protected nests. She spends about 20 minutes here before she finally moves on. This wire is simply tent pegged into the ground, which is quite simple, but so far it has prevented any predation of any of the protected nests. So after incubating in the ground for up to 90 days, the hatchling turtles dig their way out of the ground and scamper down the bank into the water. On the left, you can see some wild Bell's turtles emerging from a protected nest. This is actually the same nest that fox was trying to get into in the previous slide. The tiny hatchlings, the carapace diameter is about 25 millimeters or 1 inch. And so, as you can see, they have no trouble getting out from underneath that mesh. So over the 3 seasons, we've been surveying a 2-kilometre stretch of river, and we have found 140 rated nests, and 40% of those nests have had fox scats in or near the nest chamber. We've only been able to find 6 nests that have made it through to hatching without any protection. And we've protected 47 nests from which we know more than 700 hatchlings have emerged. And just reiterating that of those nests, of the nests we've found, more than 95% of them have been raided by foxes. So over the 3 seasons, we've collected data on nesting, the timing of nesting, location of nesting, incubation duration, and taken data on nest temperature and humidity inside the chambers. Hatching success differed dramatically over the 3 seasons. In the first season, the average nest temperature was 25 degrees and hatching success was 90%. In the second season, the average nest temperature rose 3 degrees to 28.8 degrees and hatching success decreased to 75%. That season, it was much hotter and drier, and many hatchlings perished in the nest, unable to dig their way out of the hard, dry soil. And in some cases, the yolk sac of the hatchlings had actually solidified. And then in the third season, the third breeding season, 2019-2020, that season began in drought and the river levels were low. And when we finally had rain in January, all of the protected nests were inundated by water to some degree, and hatching success plummeted to 28%. We did move a nest that was in the path of impending floodwater. We carefully moved the eggs 2 meters higher up the bank, and all of the eggs from that nest successfully hatched. So we know that hatchlings are emerging and going from the protected nests and making it back into the water. But how will we evaluate the efficacy of this nest protection as a conservation strategy? In the short term, our measure of success will be the recapture of hatchlings from protected nests. We've been surveying the Bell's turtle population since 2002, and over that 18 years, very few hatchlings or small juveniles have been recorded. So we know that the hatchlings shelter in dense aquatic vegetation and that they tend not to turn up in baited traps until they're about 2 or 3 years of age. So we expect a delay between the time of them emerging from the protected nests and them showing up in our surveys. We're just about to start our surveys in the next couple of weeks at spring in Australia, and we'll be surveying, of course, in treated and control sites. And we're hoping that the hatchlings from our first nest protection season will be starting to appear in our surveys soon. And aside from intensive surveys, we're also planning on fitting some of the newly emerged hatchlings with microtransmitters and radio tracking them to assess survival and habitat use. Our long-term measure of success will be to see animals from the protected nests survive and join the breeding population. However, we're not sure how long this will take, as it depends on how long it takes the turtles to reach sexual maturity. Current estimates suggest that female Bell's turtles do not mature until 20 years of age. However, this is based on a fairly small data set. And so we're currently working on new growth models and longevity estimates for this species. And preliminary results suggest that the juveniles grow much faster and mature much more quickly than the previous estimates suggested. And our new estimates will be based on over 500 recaptures that span 18 years. So our nest protection program is aiming to increase the number of juveniles in the waterways. However, we're also looking at some other strategies to increase juvenile recruitment, and they include artificial egg incubation and then release of the hatchlings back into the wild, the use of temporary electric fencing to keep foxes out of the nesting banks and beaches, and the provision of financial incentives for landholders to encourage them to undertake fox control, riparian restoration, and habitat protection. Some of the disadvantages of the nest protection program include the synchronous nesting of Bell's turtles in response to weather events, makes it difficult to locate and protect nests on a large scale in the small window of opportunity that we have before foxes find the nests. Of course, nest protection is also time and labour intensive. And as we found in the last 2 breeding seasons, nests that are left in situ are vulnerable to environmental extremes such as heat, flooding and drought. Some of the advantages of nest protection program include the reduced risk of introducing disease and pathogens to the turtle population, and the avoidance of behavioural alterations in hatchlings. It also allows us to collect environmental data simultaneously, and it facilitates engagement with landholders. It fosters environmental stewardship and facilitates community awareness and involvement. So we're going into our 4th breeding season, and we're cautiously optimistic that we're going to see an increase in the number of juveniles in our waterways. This project is funded by the New South Wales Environmental Trust in collaboration with Local Land Services and the University of New England in Australia. Thank you very much. David, I can't see my chat window, so I might have to get you to read any questions if there are any.
No problem. We've got one so far. Have you investigated what cues the foxes use to find nests? Is it mostly odor or is it visual disturbance? And it appears that the foxes kind of know the nests are there even if they are covered.
I think that they're using odor, and I think that because the predation of nest by foxes tends to be more prevalent at both the start of the nesting season, so when the nests have first been laid, and then again towards the end of the season when the hatchlings are when they're hatching and emerging. And in terms of visual cues, I find them very hard to find any sign of the turtles. So it's very rare that you can actually see tracks and imprints on the substrate.
Deal. Question 2. Is the loss of adult females due to fox predation at a sustainable level? And if so, could this be more of an issue with diminished future recruitment of breeding females?
I haven't investigated the extent of the predation of the female turtles. I just know that it's occurring. I do understand that the protection of breeding females is extremely important in conserving the species. However, even disregarding the predation of the female turtles, the fox predation levels on the nest predation levels of the foxes on this species is effectively negating any reproductive activity of the females anyway. So we have to take some action, both controlling the foxes and getting some animals back into the water.
All right, question 3. Are you using camera data to quantify the synchrony of emergence from the nest? It looks like the hatchlings are departing relatively synchronously.
Yes, I am using camera trap data to analyze emergence. I am, and it does look like they synchronize their emergence, yes.
Very good. With all species of freshwater turtles in Australia being pluridarians, do people in Australia perceive the Cryptodira turtles as strange exotic animals like kangaroos and koalas make folks from outside of Australia feel?
Yes, I do. I can't speak for all Australians, but yes, I do. And in fact, I'm quite astounded at Ethan hopping in the water with those snapping turtles, and having seen how they can just shoot their head out and grab things. Yeah, really strange, much stranger than a kangaroo or a platypus.
Any thought of repelling foxes as done for penguins by the famous dog Oddball?
Definitely. The nest protection program is, well, actually the Turtles Forever project is a consortium of ecologists and researchers, and there are lots of different projects happening around the Bell's turtle, and there are other strategies being investigated, including fox scaring devices, odors, and tastes and aversion chemicals in eggs and things like that. So yes, definitely.
All right. How will you differentiate protected nest hatchlings from unprotected survivors in future surveys?
I've been intercepting the hatchlings from the protected nests, and I've been marking them, individually marking them with scoot marks. I'm hoping that those scoot marks will last long enough for me to be able to determine who's from where.
Yeah, the second part of that was, is it only from observed increases in the population of young turtles?
Sorry, can you repeat the last part of that question?
The whole question was, how will you differentiate protected nest hatchlings from unprotected survivors in future surveys? And is it only from observed increases in the population of young turtles?
Yeah, so the hatchlings from protected nests are marked individually, and yes, it will be from records of recaptures.
Good deal. The last question reads, camera traps set up to record cold-blooded small hatchlings?
Yes, camera traps set up to record the emergence of hatchlings. However, they're not triggered. They're set up on time lapse because the turtles do not set off the camera traps. Yeah, so I have millions of camera trap images to go through to yeah, make my observations.
So many images. That was our last question. Thank you, Louise. And
thank you.
Up next is Peter Lindman.
Can't get out.
All right. Well, good afternoon, everybody, or good morning, or good evening, depending on where you're watching this from. I'm happy to be here to tell you the story today of the Graptomys geographica, or common map turtles, of Graveyard Pond in Presque Isle State Park in Erie, Pennsylvania. This is the record photograph, or at least my personal record photograph, of the common map turtles of Graveyard Pond on these two logs about 10 minutes before I took this photo, which was in late May of 2013. About 10 minutes before, I had scanned with my binoculars and counted 172 Graptomys geographica basking in the sun. Back in the 1980s, when the concept of biodiversity was a fairly new concept, I remember reading somebody talking about one concept of biodiversity being certain species that
Claire's like in a t-shirt in her living room. Okay. The person talking.
Certain species that act as a sort of biophenomenon. And I've always thought that Graptomys species in general can be a good biophenomenon in the right sort of well-preserved habitat. But the common map turtles of Graveyard Pond at Presque Isle State Park form a really good example of that. So I want to I want to talk today about a project that I was involved in recent years to emphasize this biophenomenon aspect of this particular species. So here's Presque Isle State Park. It's a 10-kilometer long sandspit peninsula that juts out into Lake Erie, curves around Presque Isle Bay, and all this is the city of Erie. That's a small portion of the city of Erie, which has just over 100,000 residents, with more people out in the suburbs of Erie. So it's a fairly urban area, and this is for people in Erie, this is their natural escape from city life. Here's the postcard view of my study site. Perry's Monument is this little peninsula right here, monument to the War of 1812 on Lake Erie. And just beyond it, that's Graveyard Pond, and this is Misery Bay. And the map here, that's Graveyard Pond, Misery Bay. So we're looking in that direction. I always say Graveyard Pond and Misery Bay, with names like that, it's obviously a very cheery place. But it's a great place to see Graptomys geographica. It's a very seasonal pattern of basking that they have. So sites like this come in the month of May and early June, and then things die way, way down in the midsummer, late summer, and again in late August and into September, the numbers of basking turtles go way up again. It's quite the phenomenon. Most of the turtles you see are Graptomys geographica. You get occasional interlopers like this female spiny softshell, painted turtles, occasional snapping turtles, but a lot of days, every turtle you see is a Graptomys geographica. Here's what my basking traps look like earlier this year in late May or early June. I have planks of wood that are mounted on hoop nets that hang down so that when I approach, the turtles jump off into the netting. And as you look at that photograph, yes, I did catch all 6 of these turtles shortly after taking that photo. Well, with this kind of abundance, obviously I'm not the only one who's noticed that we have turtles up at Presque Isle. So when I'm out working, I'm kind of working around people in canoes and kayaks who come over and look at the turtles and get a good eyeful. Often they'll take pictures or video of the turtles. And this is a free pontoon boat tour that goes out for about a 1-hour trip on the lagoons of Presque Isle. And they always swing by the turtle log very slowly, and people lean over the railing with their cameras taking their photographs and video. The turtles, obviously, as you can see from this photo, they're very habituated to having people around. The great majority of people at Presque Isle will keep very calm as they get near the turtles and keep a little bit of distance. And whereas you wouldn't see turtles letting people get this close to them in too many other places, in a place where people are always around, they get very habituated. By the way, Presque Isle has about 3.5 million to 4 million visitors on an annual basis. I think I constitute 50 to 75 of those visitors in any given year. And there are a lot of people in the Erie area who are visiting very frequently, but there is a very strong draw. On a busy weekend day or a holiday weekend, you can get 30 to 40,000 people out on that 10-kilometer long sandspit. Back in 2009, the previous photograph and this one are photographs that I took on Labor Day weekend, generally the first weekend of September in the United States. It's Labor Day weekend, a 3-day holiday. And so I just camped out on a picnic table and decided to watch people watch turtles for 3 days in a row. And so I saw the kayakers here and the people on the pontoon boat tour. And these people who showed up, 2 different groups, and you see the young woman sitting on a guy's shoulder so she can get a good photograph, and then the people who have scaled the picnic table so that they can pull out their binoculars and get a good eyeful of the same logs with all those turtles on them. So as I sat there during 2009's Labor Day weekend, I decided to record all the comments that I could hear from people on the pontoon boat tour and kayakers and canoers, and then people passing by on the main park road on bikes and rollerblades, and people who pulled their car over off the road so they could go over and look at the turtles. And I think the people of the visitors to Presque Isle State Park certainly regard these turtles as a biophenomenon, with some of these exclamations of great awe for the turtles that they're seeing there. So having seen this, not just in 2009, but I started my studies at Presque Isle in 1999. And by that time, I had been watching people watch turtles, at least informally watching them do this, for about 10 years. And starting in 2003, I joined the board of the Presque Isle Partnership, a nonprofit organization. And here's our mission statement. The mission of the partnership is to enhance the Presque Isle visitors' experience by developing, funding, and implementing projects and programs on the park while protecting the natural environment. So we've done some playground projects. We've done a lot of signage projects for things like the Perry Monument and the lighthouse that is on Presque Isle. And we have put out new picnic tables for the visitors. So after my Labor Day weekend observations of 2009, I went to the board and presented what I had observed, all those comments and some of the photographs that I had taken. And I said, we really should do something to enhance visitors' observations of this biophenomenon of the turtles. I consider the map turtles to be the signature species of Presque Isle. And that's a crowded competition with lots of bird species and other wildlife species. But I think the turtles draw probably more comment than anything. So within a few years' time, we were able to organize and design the building of a turtle observation deck. The total cost to this project turned out to be about $20,000. And that was back in 2012 that the main construction took place. In today's dollars, $20,000 US is about $22,000 US. Would have been a higher cost, except all the labor, including these 2 gentlemen you see in the photograph, all the labor was donated by a local industry, General Electric or GE. And I have to give thanks to one of our other board members, Joe Klecker, who has a brother affiliated with GE, and they set it up so that GE employees all summer in 2012, if they wanted to go out to the park and build this deck on Fridays, they could do that instead of going to the office. They would get paid their regular GE wages, but instead of working at GE and making locomotives and other things like that, they would make a turtle observation deck. So here it is in mid-construction in midsummer 2012. This is almost the finished project. The only thing that's missing in this photo is a walkway that extends to a handicapped accessible parking spot. There's a back behind where I'm standing to take this photograph is a boat ramp with a large parking lot. And so we put in a handicapped accessible parking space with a walkway and a ramp here so that a person in a wheelchair could go up the ramp, and one of our binoculars that's in a fixed position here is at wheelchair height, so it's wheelchair accessible. It's also very nice for small children to look through. And then we have one for adults right next to it. Right there, the turtle log, which is anchored in place with some 5-gallon buckets filled with concrete. We put up a sign on the turtle observation deck off to the side of where the binoculars are. And here I am standing with a couple of Buffalo State University graduate students, Jesse Karcher and Chelsea Moore, who did some map turtle projects on the Niagara River and came to visit. The sign tells a little bit about the species that live in the park, including, of course, the map turtle, which is the main species that you see from the observation deck. So this is now Labor Day weekend of 2018. I sat for a day and watched people use the turtle observation deck and recorded several visitors who used the binoculars or stood on the deck with their long lens camera to get shots of the turtles. Here's another young couple who showed up and used the binoculars. And here's a case where a child scaled the binocular stand set up for adults, and that leaves mom to stoop over and use the wheelchair/child level. So again, the total cost of this project, which seems to have borne great dividends now, we've had it in place for 8 years, and every year I see it being used quite a bit. A lot of people come driving straight up to where the parking spaces are and walk up to the deck, obviously knowing about it in advance. And the total cost was, in today's dollars, about $22,000. And if you think that is kind of a prohibitive price for any project that you might do in a similar urban setting where you have a good number of turtles that can be viewed, you probably could conceive of a project that would cost a lot less than that. The sign, for example, the design of that sign, its manufacture and the mounting of it was just $3,500. So if you already have a walkway near a pond or a river in an urban setting and you want to do a project like this, maybe all you need is the sign and 1 or 2 of these binocular stands, and you don't necessarily need to build the deck. So we spent a lot of money on lumber for the deck, but potentially a person interested in doing a project like this could do it for a lot less money. I'll finish up by telling you that an article about this turtle observation deck is going to be one of 17 articles in Colonial Conservation and Biology in the issue that is scheduled to come out in December. It's going to be a special Graptomes only issue, 17 articles, I believe it is, all on different species of Graptomes, including the turtle observation deck at Presque Isle State Park. Thank you, and if we have time, I'll take any questions you might have.
We'll give it just a minute to see if anything comes over from YouTube. That was great stuff, Peter. It's those populations of turtles in wetlands adjacent to parks and greenways are just a ready-made educational opportunity.
Yeah. When I first got to Northwest Pennsylvania, our local newspaper would occasionally run photos of the map turtles, but they would refer to them as painted turtles. I think everybody knows they're map turtles now today. All right, I have some questions, or one question. Do the map turtles have protected areas to nest at the park? And are there good habitats for hatchlings and small juveniles to live? Well, all of the area where the map turtles are at the park is protected as an ecological preserve. We try to keep it in as natural a condition as it can be, minus the few roads that go through the area and a few buildings here and there. So it is a natural area. As for hatchlings and small juveniles, I catch very large numbers of 1- and 2-year-old turtles and 3-year-old and 4-year-old. I have about 65 to 70% of the turtles that I have caught are with growth lines, which go up to 6 years of age in the males and up to 10 years of age in the females. So we are getting a bumper crop of young turtles every single year. I've never had a year that is missing in terms of turtles of the age that would belong to that cohort. So there certainly is some good nesting habitat. I've done a little bit of nesting studies in the past, not in recent years, but I do know where some areas are where they can nest with relatively few people around. They also sometimes nest in areas where maybe lots of people are around. Question number 2, when will the live turtle basking cam be installed so I can watch? Well, step 1 in that is to get past this pandemic, because the Presque Isle Partnership is not really able to operate during the pandemic. We do fundraisers with concerts and craft fairs and festivals on the park, and we haven't been able to do that all year. So once we get past that, then perhaps a live basking cam might be an idea. Did this effort include enhancement or installation of basking spots in areas where the public could view? Good question. I think the initial impetus came from this because a park naturalist by the name of Brian Gula back in 2000 anchored a large cottonwood tree trunk from a fallen cottonwood right out in the middle of Graveyard Pond, and the turtles took to it like crazy. And so when we built the deck, we ended up building it as a slightly different position from where we originally had planned. So we put another pair of cottonwood trunks together. That's what you were seeing in the photos today. They're anchored in place with chains and 5-gallon buckets that are filled with concrete. So they're not going to go anywhere soon. Eventually they might break up and break apart, but we can always get new fallen cottonwoods and put them in place of the originals. Do you see this as a possible model for other nature centers, preserves to apply to their sites to increase community involvement with and interest in wildlife? Absolutely. And I didn't have a chance to talk about it in a 15-minute talk today, but when the article comes out in CCB in December, I actually point to some other urban areas where I have seen good turtle populations, rather abundant turtle populations, and I could very much see some local group organizing to put up a sign and further educate the public about what it is they're seeing. All right, I think that's all we have time for. And I'll stop this and we can move on. Thank you.
Hi, everybody. Well, good morning, good afternoon, and very good early morning for those fellow Australians and others there here joining me today. It's 2 o'clock in the morning here in Darwin. I'm Carla Eisenberg, and I am based at Charles Darwin University in Darwin, which we call the top end of Australia. Just to give a little bit of a background where we live. A few hours down the road is Kakadu National Park, where back in the days, Crocodile Dundee movies were recorded. We even have one of the retired crocodiles here in Darwin that we can go and visit once in a while. They used to be in the movies. And I am based also in the Research Institute for Environment and Livelihoods. And this talk is based in research from Isabel Ellie that has done her master's research with the northern long-necked turtle, also in partnership with Sutcliffe, Larissa Schneider from Australian National University, and Keith Christian also from Charles Darwin University. So we were talking about here a little bit about wildlife contamination by heavy metals. And wildlife contamination by heavy metals from mines and other enterprises is of great concern worldwide. The concern is not only towards the animals suffering from the contamination itself, but also the communities that rely on these animals as part of their diet. As an example here, I have the Mariana disaster in Brazil in 2015. Where one of the dams for an ore mine has collapsed and all the water, contaminated water, washed away and has long-term, very acute short impacts and long impacts in the animals, and also the communities that rely on the fishes in these rivers downstream. And there were turtles there too, freshwater turtles. So the cultural importance and the role of bushmeat as a source of protein for indigenous communities, it's widely recognized. However, its role as a source of much needed micronutrients, particularly iron and zinc, has not been properly addressed. This lack of awareness might lead for us to labeling the consumption of animals captured sporadically by remote communities and indigenous communities as a delicacy and not necessarily as an essential for the community dietary needs. And I'm going to give you an example here, very close to Darwin in the top end. So this is the area, if this on the top here is the area where Darwin is. And this is the northern long-necked turtle. This is a little hatchling. Some people have them as pets. And they live in this, in many different places, but particularly in these billabongs, they're also infested with crocodiles. And this northern long-necked turtle is considered a key traditional food for remote indigenous communities in Northern Australia. It occurs in lowland waterways, billabongs, and seasonal waterlands on coastal floodplains across Northern Australia, and not just in Northern Australia, but also in New Guinea, the New Guinea island that's not too far away from here up to the north. So this species is traditionally harvested, particularly in the dry season, when it can be found buried in the dry floodplain, in the drying floodplains, and it estivates, and then you can see here they're finding one of these turtles that are being estivating. These turtles are really prized in indigenous remote communities here. They're considered one of quite tasty by the community, and I like saying that the community consider them, and that hearing what they say that they are it's very healthy. It's very good for you to eat one turtle once in a while, and coming from communities that don't have a lot of access to food necessarily, healthy food for markets. We will say that these turtles are the avocado of the bushmeat world. They are green, they are tasty, and they are full of good fat, according to the communities. And the idea of turtle harvest, depending on where it comes from, might sound a little bit odd. So turtle harvest, and it's a lot of the studies have shown it's not always sustainable. Usually turtles tend to have low juvenile survivorship, delayed maturity, and high adult survivorship. For this turtle species, a small reduction in the adult survivorship over time due to harvest can lead to population decline. This is not the case for the northern long-necked turtle. This is a relatively fast-growing and early maturing species. It also lays up to 4 clutches per year with around 12 eggs each. And Damien Ford, former research 2010 to 2013 with this species in Maningrida, have demonstrated this species population can sustain fairly high levels of indigenous harvest. However, harvest is not sustainable when there is habitat destruction and predation by introduced pigs. And just to compare a little bit with the previous talk that we have earlier, when Louise was talking about predation of nests and turtles by foxes. We don't have foxes here up north in Australia, but we do have introduced pigs, and they are the big problem here. They can find and eat both nests, eggs in nests, but also the estivating turtles. When they bury themselves and they are estivating during the dry season, the pigs can find them and eat. And you do find a lot of turtle shares that are easily to identify as predated by pig, because you can see that the pigs really crunch the shell up. And it's quite clear that that was the reason why it had died. And the importance of the northern long-necked turtle to the diet of the local communities is corroborated by the high levels of iron found on its liver and muscle. It's much higher than cow muscle, as you can see here in this figure. It's almost like, and that's a loose allegory, but I'm saying that by eating a little piece of turtle meat, it's the equivalent of eating a whole cow. And just to compare with other sources of protein that you find in the area, so these are floodplains. 2 species of fishes, such as the bony bream and the catfish, commonly found and consumed in the same area where the turtles were captured, have iron levels lower than the reporting limit. So it's not much iron in it at all. And just to highlight even more the importance of these turtles for these communities. And these are very remote communities, sometimes take days for us by car to get there. And during the wet season, probably the only way to arrive there is by plane. Anemia is a serious issue in the Northern Territory, where 50% of the pregnant women and up to 25% of the children from 0 to 5 years old are anemic. This percentage increases even more when you look at the numbers in remote communities. There is a potential to use the northern long-necked turtle to improve health locally and provide opportunities for novel traditional owned land enterprises. It's also an incentive for promoting communities to protect areas against invasive species such as the pigs and buffaloes, because once we have pigs and buffaloes in the areas, there is a population decline on northern long-necked turtles. However, the traditional owners from the Finnish River catchment, and this is a catchment 3 hours from Darwin in the Northern Territory, have reported a decline on the health of harvest turtles, as well as more frequent signs of dead turtles. This area is downstream of the now closed Rum Jungle uranium mine. This mine was closed 45 years ago, but acid and metals are still draining into the environment. So you were invited. They ask us to go there and have a look on what's happening and why this is happening. They say that the turtles are looking not as good, they're not as tasty, and also there is a lot of dead turtles. We we are still trying to find out what's happening there with these turtles, but we took some samples to analyze for heavy metals and sent to the Australian National University, where Larissa analyzed this for us. And as you can see here, the values in this figure, which are average concentrations of cadmium, lead, and copper in the liver, muscle, and skin from 5. This is the average from 5 long-necked turtles harvested in the area. And some of animals have heavy metals concentration over the maximum level recommended for fish meat consumption, according to World Health Organization guidelines for copper and lead. However, it's important to note that when these heavy metals concentrations are translated to consumption of turtle kilograms per week, a person can eat up to 3 kilograms per week of turtle meat and liver without risking their health. And when you say that to the community, they just laugh. There is no way on earth that they will eat that much turtle in a week. So the good news, at least on these really, really initial pilot studies, is that these are very good news for the remote communities can still harvest these turtles. However, this was just, as I said, a very small pilot study, and more monitoring is needed for freshwater turtles in contaminated areas, particularly species that are traditionally harvested. Similar to marine mammals, turtles have a long lifespan and occupy high trophic levels in the food web. Many heavy metals accumulate with age. Therefore, long-lived animals such as turtles, which feeds on other aquatic animals, are likely to have one of the highest concentrations of heavy metals in the given ecosystem. Most turtles are omnivorous, but the northern long-necked turtle is predominantly carnivorous. Animals high in the food web also tend to have higher concentrations of heavy metals in their body. Food is usually the main source of exposure to heavy metal. And as you can imagine, heavy metal contamination in bushmeat could have severe consequences to micronutrient availability to remote indigenous communities. And unfortunately, if harvest turtles have high levels of heavy metals, pregnant women, nursing mothers, and children should be the first ones to be discouraged from consuming turtle meat. And again, unfortunately, these are the same age groups who are most vulnerable to anemia and need this high source of iron the most. Furthermore, at least in my own experience in Amazon, Papua New Guinea, not so much in Amazon, but definitely in Papua New Guinea and Northern Australia, harvesting turtles is a women activity. And as I have experienced myself in Papua New Guinea and with those turtles in the Kikori River, the women go in big groups to harvest these turtles, and they will eat most of that with the children. So, and we are also, now that we noticed that the levels of iron were really high on the northern long-necked turtle, we have checked the iron level, levels in pig-nosed turtles and a few of the Podocnemis in Amazon, and they all have the same, similar, very high levels of iron. So it's that the fact that these turtles, even though they are not eaten frequently and just once in a while, they might be giving that iron boost that these communities need once in a while. This is just a hypothesis, and we still need to test it. But as we are checking more and more turtles, most of them have high levels of iron. I'm not saying here that everybody should go out there and start eating turtles for their health, but I'm just trying to point out that these turtles are very important for the dietary needs of indigenous remote communities. And there is very little heavy metal monitoring of bushmeat consumed by indigenous communities in Australia and other remote regions, such as Amazon and Papua New Guinea. If anemia is to be halted in remote indigenous communities, there is an urgent need to better understand the role of bushmeat, such as freshwater turtles, as a source of micronutrients, and how pollutants could endanger their population and those harvesting them. And I would like to thank the communities and groups that have welcomed us in their land to do this research. The Twin Hill Aboriginal Corporation, Finniss River Floodplain Traditional Owners, and also the Finniss Reynolds Stakeholder Group, as well as LaBelle Station and Bulgur Ranges and Ironstone Lagoon Nurseries, and our partners, the Territory Natural Resource Management, Northern Land Council, and the Australian National University. And also my youngest one, Taiko, that's in this photo, that went with me in the field in Crockett's infested water safely, a bit far away from the water, but joined us in our field trips when he was around 8 months old. Thank you, Anna. I'll take any questions now if you have. Sorry.
We do have 2 questions in the chat. Would you like me to read those off to you?
Yeah, the chat has, I think, oh, I found it. The question 1, will providing mineral supplements to local communities be more directly benefit to their health status without risk heavy metal exposure? I think that's something that needs to be taken account. If harvesting traditionally it's not sustainable, or the turtles are actually with heavy metals and levels that are problematic. And I think another option is also to introduce vegetables and greens in the community gardens that are high in iron. And this is very important, if particularly in conservation projects such as, and that we're thinking here about, for example, the pig nose project that Pico project that Helen was describing previously. We need to tackle the fact that half of that population is anemic, and how and provide them with alternative sources of iron, which in a long term, probably introducing more iron-rich vegetables and greens in their gardens could solve, before we can tackle the problem that the turtles are being over-harvested and they are endangered. And the second question, do locals prefer harvest long-necked turtles over hunting wild pigs? And is there any farming opportunity for them, just like the Chinese soft shell in East Asia? Yes, that's also a great question. Yes, so turtle is very, very well sorted meat. If you have turtle or any other type of meat in this remote community, you go for turtle. That people love it. That's why I joke it's the avocado of the mush meat word. It's just people love them, and they do hunt and try to keep there is pig hunters here around too, that and they and also there is programs where people try to decrease the number of feral pigs by shooting them in helicopters and things like that. And that's something that we are trying to look into now, is how effective this is for the populations of turtles. But pigs, wild pigs, are not necessarily, especially the big ones, they're not really good for eating. They don't taste good. So it's not really an option as to stop eating turtles, so people just eat pigs. But there are alternatives on how people can manage the pigs population so we can have more turtles, and that's definitely something we are looking into. And I think that farming opportunity, it could be, but an option, or just but things here are very remote, so having a farm and having something that you have to keep constantly fixing and getting materials to fix, it's quite hard, especially during the wet season. So although it is an option, definitely an option, maybe something like free ranging, where you protect a billabong against all the threats such as pigs, buffaloes, and keep the area healthy so you can harvest more turtles. It's an option, but also farming can be an option, and that's why I meant by Indigenous-led enterprise. This could be a source of economic benefit to these communities. The third question is, have the traditional diet of these communities changed over time? And do you expect turtle consumption to decline with new generations that have the tendency to move away from their traditional cultures? There is a little bit of that. And one of the problems that we have in these remote communities that usually if you decide just to go for market food, so you go to the only store that, if you're very lucky, you have 2 stores in your community, the type of protein you're going to get there, it's fried chicken, it's terrible, it's not good for you. So there is health can become because of poor diet. It can be a big problem here in remote communities. So actually there is a movement of trying to bring the kids back to country, back to the traditional ways of looking for bush food and looking for the food that's available in the environment, because it's much healthier for them to collect this food and eat this food than eat the food that's available in the stores. And I think those are all the questions that I have today. Thank you very much.
Okay, well, I'm going to go ahead and get started then. So thanks everybody for being here. So my name is Adam Claus. I'm a postdoctoral researcher at the Natural History Museum of Los Angeles County here in California in the USA. And before I get started into the actual meat of the presentation here, I do want to acknowledge my 2 co-authors on this project. So Aaron Celestian and Greg Polly, who are both curators here at the museum, and both of whom have contributed in really big ways to the development and implementation of this project. So with that, let's dive right in. So there we go. So environmental plastic pollution is this really big, widespread problem that I'm sure many of you are aware of. And one of the principal ways in which this affects animals is when animals inadvertently or intentionally, in many cases, eat environmental plastic. So once ingested, this plastic can often accumulate in the stomach or even block the gastrointestinal tract or the stomach, which can then lead to the death of the animal, as shown in this picture here of a young albatross. Additionally, even if the plastic does not kill the animal, it can also lead to sublethal effects, for instance, by displacing food items and leading to poor nutrition of that animal. And so this is a problem that's especially well known, as many of you in the audience already know, in marine turtles. And so marine turtles eat a wide variety of different types of plastic in the environment. But perhaps most well known is this consumption of plastic bags, which both leatherback turtles and to a lesser extent green sea turtles confuse for their jellyfish prey and ingest it in that way. But one of the big questions, right, is this a problem? You know, it is a problem for marine turtles, but is this a problem for freshwater turtles? And so at this point, we're not really sure if this is something that we need to be worried about for this highly imperiled group of animals. And we'd like to better understand what the situation is in the current state of knowledge. So for the remainder of this talk, I'm going to be sort of exploring what we do in fact currently know about plastic ingestion in freshwater turtles. And I want to conclude with sort of a call to action to the non-marine turtle community to try and better understand this situation and evaluate the potential for this possible threat. So to figure this out, we took two approaches. So the first thing that we did was we conducted a literature review. So we queried Web of Science and then supplemented those search results by querying the natural history notes published in Herpetological Review, the journal. And what we did with the studies that we returned is that we wanted to sort of look at two specific things. So the first is that we wanted to identify all turtle diet studies, both marine and non-marine species, published since 2014, in order to compare relative research investment in these two groups to determine if there was an imbalance in that research investment or not. The second thing that we wanted to do is identify all non-marine turtle diet studies published roughly since the 1920s to fully evaluate the number of studies that have currently documented plastic ingestion by these turtle groups. And so I'll just note that we excluded studies that lacked the potential to document plastics, so studies that only focused on stable isotopes or eDNA, and instead only considered studies such as stomach flushing, gastrointestinal tract dissections, or fecal analysis that had the potential to document plastics. So that was the first thing we did. And then the second thing we did was an empirical study to look at how you actually go about collecting data on plastic ingestion in turtles and teasing apart some of the potential problems that you can come up with when trying to do these sorts of studies. And so for this empirical aspect of the project, we focused on the UC Davis, or the University of California, Davis Arboretum in Northern California. And so that's shown on the map here. That blue squiggly line is the waterway, the Arboretum waterway. And so what I want you to take away from this map is just that the waterway, the surroundings of the waterway are quite urbanized. And so both the city of Davis and the University of California, Davis campus both discharge wastewater and also runoff into the Arboretum. And so this is a source for potential plastic contamination in this system. So in the Arboretum waterway, we captured 65 individual non-native red-eared slider turtles, Trachomys scripta elegans, across a suite of different age classes and including both adult males and adult females. And after euthanizing these turtles, we then dissected their stomachs and their intestines to search for potential ingested plastics. Now our search methods only enabled us to identify plastics that were greater than about 0.5 millimeter in size. So we were really only looking at macroplastics, although we could detect some of the larger microplastics in this sample. And then we took the very important step, as I'll mention a little bit later in the talk, of verifying those plastics using this quantitative tool, Raman spectroscopy. And the last thing I just want to highlight really quickly, and just keep this in mind because I'm going to come back to this point a little bit later, is that in the Davis Arboretum, the red-eared slider population actually co-occurs with a native population of the western pond turtle, which is an imperiled species of conservation concern. And so interested in potentially using red-eared sliders as a proxy for the plastic situation with these imperiled western pond turtles. All right, so what did we actually find? So let's focus on the literature review first. So when we look at the available literature that's been published on turtle diets since 2014, what we find is that marine turtles constitute approximately 47%, or nearly half, of that literature. And so this is despite the fact that marine turtles only constitute about 2% of global turtle species diversity. So a very strong imbalance in terms of research investment in marine turtles relative to non-marine turtles in that regard. And then another way to sort of look at that is that since 2014, On a per species basis, you've got approximately 8.4 diet studies published on marine turtles versus only 0.2 diet studies published per species of the non-marine turtles. So again, we can't necessarily say that plastic ingestion is or is not a problem in non-marine turtles relative to marine turtles per se, but on a per species basis, we have very little understanding of the effects and whether or not non-marine turtles are actually ingesting plastics or not. So warrants further investigation there for sure. Just to dig into this a little more deeply, so again, just sort of emphasizing that imbalance in sort of a slightly different way. So if you look at marine turtles, all 7 recognized species, or 100% effectively of marine turtles, have been reported ingesting plastic. And in many cases, there are quite literally dozens of studies on particular species of marine turtles documenting ingested plastic. But if you then look at non-marine turtles, again, only 5 of 351 species have been reported ingesting plastic. And even if you expand sort of the scope out to include non-marine turtles that have ingested non-plastic litter, so things like cardboard or glass, you're still only getting up to 8 of those 351 species for which that has been reported. So a very large discrepancy there in comparing marine versus non-marine turtles. And then I want to show you this table really quickly. Don't worry about trying to take too much away from it, but basically every row in this table illustrates or represents the 15 studies that we have identified so far that document a non-marine turtle species eating either plastic or non-plastic human litter. And so again, if this was a marine turtle table, this would be hundreds of rows long. So just again, further emphasizing how little we know currently about this plastic ingestion problem in non-marine turtles. You'll have another chance to look at this table a little bit more thoroughly later in the talk, but for now I want to switch over to that empirical study that I mentioned highlighted here in red and build that out a little bit in terms of those results. So what did we find with that empirical study of red-eared sliders in the Arboretum? Well, the basic takeaway here is that red-eared sliders in that system do in fact eat plastics. The twist comes when you actually start talking about plastic ingestion frequency or how often they're eating plastics. And so that twist relates to the fact that true plastics are very easy to confuse with plastic mimicking organic material like crayfish exoskeletons. So just drawing your attention to this figure on the right-hand side here. So on the left-hand column, so panels A through D illustrate plastics that we pulled from the stomachs of a red-eared slider that are in fact confirmed verified plastics versus the column on the right-hand side. So panels E through H illustrate materials that we believed visually to be plastics, or at least potential plastics, but which our Raman spectroscopy method confirmed were actually not plastics, but were organic materials. And so this is really important for understanding what the true frequency of plastic ingestion is you have to be able to identify confidently what is and is not plastic. And so if we had just used our visual techniques, we would have had an 11% plastic ingestion frequency among that population, versus once we actually verified the materials, that dropped to 6%. So a pretty big change there. So just to give you a sort of a deeper look at what this Raman spectroscopy output looks like, I wanted to show you this really quickly here. So what these two panels are illustrating are in the red lines. So the red lines illustrate Raman spectra for known reference plastics, whereas the blue lines in each panel indicates the Raman spectra for a suspected plastic sample that we pulled out of a turtle stomach. And so the key thing to note when you're interpreting these graphs is you're looking for peak matching. And so as you can see, the peaks in the red lines very closely match the peaks in the blue lines, indicating that those blue lines, AKA the turtle stomach samples, are in fact plastic. So polystyrene in the case of the panel on the left, and polyethylene on the right-hand side there. So Raman spectroscopy is a really great tool for quantitatively verifying plastic, and is really something that we view as being quite important. All right, so based on these results, so sort of this literature review, and then looking at sort of the nitty-gritty of how you go about doing these studies, we do want to issue a call to action to the non-marine turtle community. So non-marine turtles are clearly ingesting plastic and non-plastic human litter, but this is something that's quite understudied, at least relative to the marine turtle literature. And so plastic and non-plastic litter ingestion certainly could threaten non-marine turtles. And we believe it potentially has a pretty strong threat, particularly in urbanized areas. But this is something that's going to require a lot more research investment on a global scale by the community before we can make really informed decisions about if this is something we should be worried about or not, and where we should be worried about it potentially. And so for those of you who are thinking about potentially getting involved in research along these lines, we do want to offer 3 sort of takeaway recommendations that we hope people will consider as they're beginning to explore the possibility of undertaking a study of this type. So the first thing we want to emphasize is that we'd like people to at least consider the use of non-native turtles as a proxy for plastic ingestion by imperiled native turtles. So remember, in the Davis Arboretum, we were looking at specifically the red-eared slider, which was non-native and which has been introduced widely throughout the world to get a better sense of what the potential problems were with respect to plastics of the imperiled western pond turtle. And we think that that study design is something that could be duplicated in many places across the globe, given that these plastic detection techniques are often somewhat invasive and may not be something that's advisable in native turtles of conservation concern. So using a red-eared slider or another non-native species to try and get at that question is something that we think is useful to consider. Secondly, we highly encourage the community to always flag studies that include plastic ingestion data by including terms like plastic or litter in their study title and or their study keywords. In this way, we can ensure that the scientific community can actually discover these studies and that they don't go overlooked, which is going to be really important moving forward. And then lastly, we strongly encourage people studying this to verify the ingested plastics that they document with one of several available quantitative tools. So Raman spectroscopy just being one option that's out there. Because it really is difficult, especially when you're dealing with tiny fragments of plastics or suspected plastics, to know for sure if you are in fact dealing with a plastic or if you're dealing with a plastic mimicking organic. So with that, I just want to conclude really quickly by sort of soliciting all the viewers that if you do not see your non-marine turtle, plastic, or litter study listed here in this table, please do reach out to me. We would love to hear from you and we want to cite your work. If you have unpublished observations or data sets on turtle plastic ingestion, non-marine turtle plastic ingestion, we would also love to hear from you and sort of discuss your data set in the context of what we've been finding. And so I'll just leave this up here for a few seconds, but we'll be happy to take any questions at this point. And David, I can't see the chat, so I'm going to have to ask you to read questions.
Absolutely. We do have one question so far.
Okay.
Was there any indication from your study at the Arboretum that there are negative effects from plastic ingestion? Differences in BMI, for example?
Great question. So no, we do not currently have data suggesting that the plastics that we documented were having negative impacts on the specific turtles in which we found those plastics. And so that's certainly not to say that plastics couldn't be having an effect. So our sample size, as you saw, was 65 turtles, so relatively small. And one could also presume that if ingested plastics were having an effect on a turtle, that turtle could potentially die quite quickly. And so that would make it difficult for us to detect those sort of lethal effects of plastic. And obviously we did not test for sort of those small scale non-lethal effects, stressors of that plastic ingestion. But at least in that system, based on the data that we have, we don't have strong evidence to suggest that plastic ingestion is sort of a population level threat to those red-eared sliders.
All right, we're going to hang on just a little bit longer. There is question 2. It just came in. What is the cheapest method to verify plastic and gut content?
That is a good question, and I don't know that I can answer that in a very clear way. So here at the museum, we were really fortunate in having a Raman spectroscoper here that we could use essentially for free. And so given that we had that resource, we sort of jumped on that and used that resource that was available for us. So I didn't do a cost analysis of all the other potential methods that are out there. But there are, I would just emphasize, a diversity of different methods that are available. And so depending on your specific context, potentially even just collaborating with someone would be potentially a way to run those samples. It's really just a matter of having the equipment and then having someone who knows how to run that equipment. So there's very little costs aside from that time and expertise, at least with respect to Raman spectroscopy.
Very good. Question 3.
Great.
Correlation between the probability of having plastic in a turtle and its size or age.
So with respect to our specific red-eared sliders from the Arboretum, I'm assuming is what the question is referring to. So we had plastics identified both in juvenile red-eared sliders and an adult female red-eared slider. So we did not see ingested plastics in the yearling turtles. So we had 29 yearling turtles that we dissected their gastrointestinal tracts. So again, a relatively small sample size, but not finding plastics in those really tiny turtles is also somewhat explainable by the idea that, if they're very young, they just may not have had the chance to encounter and ingest plastics. But that's really just speculation on our part. So juveniles and an adult female we documented in the Arboretum with plastic.
All right. Do you think that this is target or non-target ingestion? Are the freshwater turtles ingesting due to attraction to the plastics or incidentally due to widespread presence throughout the waterway?
So we don't really know is the short answer. I think it could plausibly be either explanation or a combination of the two in terms of explaining why specifically red-eared sliders are ingesting plastic in that system. Without behavioral observations of sort of following turtles and getting lucky to see what they're eating and where and observing actual ingestion of the plastics, it could be difficult to evaluate that. One thing that we would like to see is, and one thing we did not do in our study was compare plastics ingested by turtles with environmental plastic samples, which would allow you to potentially evaluate if there's any sort of selectivity or evidence of selectivity happening. And so we're just starting to see some of that evidence come out with the marine turtle literature. So there was a recent paper showing that green sea turtles, which often feed on green seagrass and other green vegetation, that they will actually preferentially take green colored plastics in the marine environment. So it's entirely possible that there's similar sorts of selectivity going on in non-marine turtles, but we just don't have the evidence yet to be able to say that. So I would encourage people to investigate that. It would be great to see that data in the future.
All right, last question.
Great.
Do the types of plastic found most in turtles, are they floating plastics or would they be plastics that would sink to the river bottom?
That is a really good question. I don't know. I can take a closer look at what the specific, what's hard is that for the pre-existing literature on plastics, often it's very non-specific. So dietary items are simply identified as plastic with no further detail. So in terms of looking at previous studies, it's sort of impossible to answer that question with published data. In our specific case, we documented at least 3 different types of plastics, so polyethylene, polystyrene, and nitrile. I'm not sure if any of those would sink or float in water, but I can certainly investigate that. And I think that may be something that we want to include in our paper on this. So thank you for the question. That's a good tip for something for us to look at.
Thank you very much, Adam. I'm sure we will see more on this subject in the future.
Sure. Thank you guys very much. Appreciate it.
Okay, great. Hello, everybody that is out there listening and watching. Thank you for being here today. My name is Maris Brunwhite. I am a wildlife veterinarian and epidemiologist with the St. Louis Zoo Institute for Conservation Medicine here in the United States. And I'm going to talk to you today about some field veterinary research I recently did looking at hematology and biochemistry in radiated tortoises. And before I get started, I just want to, of course, acknowledge my co-authors on this project and really say that this was a collaborative project between TSA Madagascar, the St. Louis Zoo, and the Wildlife Conservation Society. And I'll be thanking individuals at the end. So to put this in context, a lot of you, I'm sure, are familiar with the radiated tortoise story, but radiated tortoises are endemic to southern Madagascar and are currently listed as critically endangered in the IUCN Red List. The primary threats that they are facing are habitat loss and illegal collection. And by illegal collection, we mean both food and on a much larger scale right now for the pet trade. And the story that probably many of you are most familiar about and that one of my co-authors, Dr. Niaina, spoke about earlier in the symposium are these mass confiscation events where thousands of tortoises are confiscated at one time. And what that means is that TSA is now caring for over 24,000 radiated tortoises. And this makes building a robust confiscation to release pathway a key part of the conservation plan for this species. Blood work is an important component of building a robust confiscation to release pathway. Hematology and biochemistry are foundational measures of health. So this is the basic blood work that you get at a doctor, but just tailored for a tortoise. We can use this at triage. We can use it to treat sick animals. We can use it during pre-release health assessments to make sure we select only healthy animals for release. And we can use it during post-release monitoring to look at success of those animals. What we are faced with many of the species that we deal with is not knowing what is normal for either the species, a given age class, a given population, or a given technique. We know that there's a lot of variability in these parameters across each of these dimensions. And while we have reference ranges available for radiated tortoises, the current ones available are either on very small sample sizes, so 18 animals, or they are across a variety of techniques. So they tend to be very broad and they're all based on captive animals in North American institutions. So not a great tool for comparison for this confiscation to release pathway. So to build this foundation, we set out with the objectives to establish hematology and biochemistry reference intervals that would be useful for this pathway. So that is based on clinically healthy, previously confiscated radiated tortoises living in Madagascar. And in this case, it was subadults because those are the ones being evaluated for release. And then secondarily, we were interested in looking at differences based on venipuncture sites. So that's subcarapacial or brachial. I'll go into that a little bit later. 3 different very common ways of counting white blood cells: the Nat-Herrick method, the Leucopet method, and a blood slide estimate. And then getting a little glimpse at population differences, looking at our population, this TCC or TSA population versus free-ranging tortoises nearby. So in order to accomplish this, I traveled along with veterinary staff from WCS and other staff from the St. Louis Zoo to TSA's Tortoise Conservation Center in southern Madagascar this past February. And all of us were there to perform a pre-release health assessment on a little over 1,000 radiated tortoises. And we used this opportunity to do this research by sampling a subset of 120 clinically healthy tortoises. This wasn't a truly random sample. We did the first 5 to 18 that we found in the enclosures each day so that we could standardize time of day. These are all subadults, so they're unknown sex. And every tortoise received a physical exam, weight, morphometrics, swabs for PCR for primary pathogens of concern, which I'm happy to say have all come back negative. And they all received a blood draw either from the subcarapacial sinus, which is, you can see me drawing blood in the picture. It's right above the head, sort of right under the carapace, or the brachial vein, which is in the armpit. And we also have the opportunity to sample a very small number, about 20 tortoises outside the fence. So this is in native radiated tortoise habitat. So we just went outside and were able to find free living radiated tortoises out there. Again, we did the first 10 we found each day. We confirmed they're all clinically healthy, but a really important difference is that this is all age classes and sexes. So very different than the TCC sample in that way. We went through identical procedures, and then all samples we collected were analyzed or fixed on the same day in the field. So for the specific biochemistry and hematology methods, we used the sort of quote unquote portable VetScan analyzer. This is made, the rotors that we used are made for reptiles. You can see the analyzer it's about the size of a big water bottle, but it doesn't like heat. So we had to pad it with ice packs to keep it working. And a refractometer to measure total solids, which is a simple handheld device that's commonly available. For hematology, we measured packed cell volume just by spinning down hematocrit tubes. We looked at the white blood cell differential, which is the percentage of different types of white blood cells in each sample. And you do that by just looking at a slide and counting. And then we did the 3 different methods of white blood cell quantification or counting, which again is the Nat Herrick, the Leucopet, and the slide estimation, just looking at that blood slide and counting using a diff quick stain. And I just owe a huge debt of gratitude to Jane and Dahlia, the technicians from WCS and St. Louis Zoo, who did all of that reading. It was a ton of work. So what we found when we compare these results that we got in our current reference intervals from these TCC animals to prior published or available ranges, we can't do statistical comparisons between all of these because some of the prior ranges are just sort of a median and range presented, but on average, many of the values were similar, but mostly were more precise. So we were able, with a larger sample size, to do what we wanted to do, which was to hone in that range. And then a few of them were lower or higher, and I'll pull these out for you. So total protein was lower, and potassium and sodium were higher than we would have expected compared to prior ranges. And these are known artifacts of the analyzer that we were using. So in other turtle and tortoise species, they've found these exact same things when comparing the VetScan analyzer to a gold standard analyzer. So important to know what analyzer you're using. And the other thing that was lower is calcium and phosphorus. And this is also to be expected because we were looking at subadult animals, whereas adults, female reproductively active adults, are going to have a much higher calcium and phosphorus to build those eggs. And we did find a few significant differences by venipuncture site. So this is just in the TCC population. So all of the values that were different were lower in subcarapacial samples than brachial samples. And they kind of cluster into 2 groups. So that first group is hemolysis, which is the rupturing of red blood cells in your sample. And that's usually related to your technique. And then AST and creatine kinase, which are also associated with hemolysis, but also associated with muscle damage or tissue damage. So AST and creatine kinase were still significantly lower in subcarapacial samples, even when I controlled for hemolysis. So this is most consistent with the restraint and the type of tissue damage, which is an extreme term for what I mean, but it's a slightly more traumatic technique, at least in my hands as the person drawing this blood. The other group was uric acid, glucose, and phosphorus. And the most likely reason for this would be either lymph or cerebrospinal fluid dilution in the subcarapacial samples. But it's a little puzzling to me because we would also expect the packed cell volume and the total proteins to be lower in lymph diluted samples. So I've got a little question mark on that one. I'd love to hear if anyone has thoughts on it. And getting to the white blood cell quantification methods. So you can look at these reference intervals grossly. These were the exact same samples from the exact same animals, and we got very different results. So we have some pretty significant clinical disagreement as well as statistical disagreement between these methods. So to look at this in more detail, I put together Bland-Altman plots, which just plot the means of paired samples against the differences. And we consider the Nat Herrick a gold standard. That's sort of field standard at this moment. And what we find when we look at the slide estimate is that it is basically overestimating or has a constant bias of about 4,000 cells. So you can adjust for that a little bit in your expectations, but you can also see that when we get to high counts on the right-hand side of that graph, the variation is pretty significant. And even in the middle, there's a good amount of variation, but at least it tracks sort of consistently across. Then when we look at the leucopet versus the Nat Herrick, we lose almost all agreement. We really want to see these dots in between those two lines, and we want the green line and the red line to be really close together. And in this case, they're positive 10,000 cells and negative 10,000 cells. So when I investigated this further, this seems to be an artifact of the fact that leucopets don't count lymphocytes directly. They use calculations where they infer the lymphocytes. So the higher percent of lymphocytes in the sample, the greater the disagreement between the leucopet and the Nat Herrick. So that seems to be where that source of error is. And lastly, this is really just a qualitative comparison between the TCC subadults and the free-ranging tortoises. But again, everything that we found in our TCC adults was lower than that in the free-ranging tortoises, and the ones that were clinically significant in their differences were packed cell volume, total solids, total protein, and uric acid. And what this is most consistent with is really just hydration status. So that's a reassuring result. It's positive for the TCC tortoises that they're very comparable to the wild tortoises, except for hydration, because they have really quote unquote unnatural access to water. They have water supplemented regularly and have a very wet diet compared to the wild tortoises. So our takeaways from this are really that we did find clinically significant differences, as well as statistically significant differences where we could test for them between different populations, the different venipuncture sites, and different testing methodologies. Some specific things to highlight are that hemolysis and tissue trauma might be greater with your brachial samples. Lymph or CSF dilution may be greater with subcarapacial samples. The VET scan has some predictable errors and limitations, so it is just reinforces that idea that you want to know your analyzer when you are evaluating results. And really importantly, the leucopet looks like it could be a very poor choice in high lymphocyte species, so species that have a lot more lymphocytes than other white blood cell types, which is the case for the radiated tortoise. So all of this together really says to use technique-specific and population-specific reference intervals whenever you have them, and to know your differences in case you don't have that reference interval available. And finally, I just want to thank the incredible team of people who did this with me. I'm just going to go left to right really fast. So it's Crystal, Dahlia, Tony, Jane, Niana, Rihanna, Mena. We don't have Fonsua, who is the other keeper who gave us a huge amount of help. And then circling back around is Bonnie Raphael, and I had to put Paul Kelly in there because he wasn't there when we took this picture. So this again was a great collaboration between TSA, the zoo, and St. Louis Zoo and WCS. And I also want to thank Abaxis for loaning us the VET scan analyzer that we used on this trip. With that, I will take any questions. I'm just going to see if I can get my chat open.
No worries. We're going to hang on just a few more seconds and see if we get anything over from YouTube.
And you know, David, I'm not seeing, somehow my chat isn't opening now that I have my share screen open. So I'll have to have you.
Not a problem. Not a problem. We'll hang on just a little bit longer. Thank you for your presentation. And while we wait for a minute, coming up next is going to be Patrick Baker, talking to us about seasonal changes in algae coverage on the carapace of painted turtles and what the implications are for the symbiotic relationship.
And just quickly, also, Adam, if you are still listening, I just have to say that everything I know about red-eared slider anatomy and physiology is from red-eared sliders from the Davis Arboretum, because that's where I went to vet school, and that's how we learned like all of our reptile medicine. So it was an interesting talk to hear.
And we are extremely grateful for the efforts provided by you and the St. Louis Zoo and everything that you guys did.
More than happy for you.
We do have one question or two questions coming in here. Do your results cast any question about the usefulness of pre-release assessments?
The usefulness of what? I'm sorry?
Pre-release assessments.
Oh, I would say no. And I'm wondering if the question is because we found basically normal results. I think I can say that we had a couple of animals that we excluded based on physical exam. And when we looked at their white blood cell counts, they were very high. So that actually is not really part of what's going to become the published results of this, but it verifies to me that checking those animals before they're released is a worthwhile endeavor. It's a little bit different when you're about to release 1,000 animals versus 5 or 10 or 20. When you are down to really small numbers, the health of each individual really, really counts. So I would say it's probably more important in that scenario.
Good deal. So which BC count would you recommend? Is NH hard to do in the field?
Yeah, so I could give a totally separate talk about which techniques I think are helpful and why. And I thought a lot about that when I was putting together this talk. So if I would say the Nat Herrick method is, when you look at it on a graph, it is the best in terms of consistency. And so if you, again, care a ton about the health of a given animal, or you are doing it for research purposes, I would bend towards the Nat Herrick method at this moment in time. If you are monitoring a patient over time for some reason, I would still just do a slide. You need a drop of blood to do a slide, and you get a ton more information from it, and you can do it much more quickly and with much less training. So, in a perfect world, I would probably always do Nat Herrick and a slide. So I have all of that information, but the Nat Herrick method is, you can definitely do it in the field, but it is way more time consuming, and you really need to learn the method well. And I would not do the leucopet for this species based on what we've seen here, and what we've seen here is consistent with a couple of other studies at this point. That's really looking like it's not the best method for this species.
Excellent. Well, that was the last question there. So
okay.
It was great work, and thank you again so much for your collaboration.
Absolutely, thank you.
Next up is Patrick Baker, West Point.
Thanks, Dave. Am I good to start?
Absolutely, go for it.
Okay, I'll also throw my video on there as well. That's not too intrusive. Okay, so thank you everybody for sticking around to the end of the session today. I know it can be a long day. I will be talking about some work that I've done in collaboration with Nathaniel Sheehan, Major Nathaniel Sheehan, colleague at West Point, and local high school teacher, Mr. Michael Mallon at James O'Neill High School, on seasonal coverage of algae in painted turtles. And on the screen right now, you can see a carapace of a painted turtle. And when I talk about algae, I'm talking about this filamentous algal growth on the carapace. And I was delighted to see in some of the other talks today, algae that wasn't the subject of the talk itself, but in the Elucer talk or in the Kelly's talk and in the alligator snapping turtle talk, at least one of those images, and in some of those cases, multiple images, you saw algae coverage on those shells, even though it's not the focus of what they're talking about. It highlights the point that it is a global phenomenon that is found in most of the families of turtles that spread around the globe. And so this algae relationship is not specific to painted turtles. So I think we can generalize from there. So the textbook relationship between algae turtle is a symbiotic relationship, and symbiotic relationships can take on many forms. And all of the forms have been proposed for an algae turtle relationship. So one symbiosis is a parasitism, where it's a positive for the algae and negative for the turtle. This has been observed most recently in the in the mud turtle. Genistern flavescens, the yellow mud turtle out in Texas. I know the group with looking at mud turtles in cattle ponds has shown that the algae can have a parasitic relationship damaging to the carapace, an entryway for disease into the turtle. We don't observe that in our painted turtle population. Another relationship that's been observed is that it could be a positive for the turtle and a negative for the algae, and that is the case where it's herbivory. Some turtles are herbivorous, and they've been known to either within a species or among species consume the algae off of each other's shells. We didn't observe that in this study, but it wasn't the focus of what we were doing either. Another relationship could be beneficial to both. So a mutualism where the algae benefits and the turtle benefits. In this case, there's evidence for that. The algae gains a place to grow and gather nutrients. The turtle could also be gaining a benefit from either eating the algae, or in the case of some carnivorous turtles, they use this algae as a lure to bring prey species closer. And then another benefit has been hypothesized that this acts as a camouflage. And then the fourth possibility is a commensalism, where there's a benefit to the algae, but the turtle receives neither a benefit or a negative interaction. And so there's evidence for this as well in the literature. Acknowledging my co-authors, Major Sheehan on the left-hand side was in our department for the last 2 years. He's just recently left and he'll be deploying. On the lower right-hand side is my colleague, Mr. Mallon, and another faculty colleague, Charles Ouellette. And then I'm in the upper right-hand side. COVID slowed us down, but it did not stop us from going to the field. So with proper physical and social distancing, we were still able to capture data this summer. And not this summer, but in many years, we've gained a benefit of having field assistance from students in Mr. Mallon's AP Environmental Science class. This is one of his groups, and they're usually found on the side of the road measuring turtles and helping to assess algae coverage. So getting into the meat of the study, at our study location, we have a lot of oligotrophic lakes. They're low in nutrient. And the question is this good turtle habitat? And we've answered that by catching lots of turtles in here. So this is good painted turtle habitat. We also catch snapping turtles and stinkpot turtles in here. So this is good turtle habitat. This is also good turtle habitat, maybe better turtle habitat. This is spatially very close. You'll see in a future slide that these two ponds are next to each other. And this one, we tend to catch larger turtles. I haven't quantified this, not larger numbers of turtles, not larger turtles. And we catch many turtles when we set our traps on this side of the road. And as you have noticed, there's a lot of vegetation on that side. It's undergoing the process of eutrophication. There's excess nutrients going into that pond, which acts as a fertilizer to grow plants, including algae. But we see lots of lily pads and duckweed and all sorts of plant material on one side that we don't necessarily see on the other. Spatially, the two ponds are adjacent to one another. So the eutrophic pond is called upper pond on the left-hand side, and the oligotrophic or very clear pond, nutrient poor pond, is on the right side, and I'll just refer to that as lower pond. And the sources of nutrients could include, there's a riding stables in the upper part of the area, and this is a capped landfill on the left-hand side of it. So both of these could be potential sources of nutrients that drive the processes that we observe. And numbered from 1 to 8 are sampling locations that you'll see in the next graph. So we've gone in and grabbed water samples and analyzed them for nutrients to answer the question, does the concentration of nitrate, a nutrient that leads to eutrophication, vary in time and space? And oftentimes I'll pose a question up top and I'll answer that question for you. So does nitrate vary spatially? Yes. We see changes in nitrate from the inflow. That was the stream where water's coming into that upper pond, and it decreases with a little bit of a rise as it goes into the outflow. Does the algae turtle relationship vary spatially? We can also say yes to that. So our results from trapping, we have 2 sample dates here, June and July of 2019. If you look at lower pond, that's the oligotrophic pond, it's lower in June than it is in July. And in upper pond, it's lower in June than it is in July. But in both cases, especially in July, it's higher in the eutrophic upper pond than it is in lower pond. So we have evidence that there's a spatial relationship, and we can start to connect it, as you'll see later, to the nutrient conditions. And just visually, this is what it looks like, a turtle that you catch in lower pond. I would guess that this is about 30% on the left-hand side and probably 70% on the right-hand side, showing just across the road from one another how different the turtles are when you have them in your hand. And so we can ask the question, is this condition consistent year-round, or does it change? That initial slide we said showed that it's different in June and July. And we've sampled further to show that in the early spring, turtles typically don't have algae on their shells, or they have a very low amount of algae on their shells. It peaks in July and then starts to taper off through the summer, but it does not completely go away by our last sampling date in October, but we don't sample through the winter, and so we're not detecting what's happening there, but we know when we catch them again the following spring, we've done this for 3 years now, that algae coverage is low again. If we compare this to the only other study that I know of that looked at seasonal changes in painted turtles, we can compare to data from Whit Gibbons, did a study in Michigan in the 60s, and I gave his graph the colors of the University of Michigan. So turtles, the blue line with the yellow dots is the Michigan data, and the orange line with the gray dots is our data. We can see a similar pattern. They've got more sampling dates, and I can say that Whit's data I pulled from text and made this chart. But by comparison, we can see that there's a peak in July is the same that he reports, and a taper into October, but not a complete removal of algae by the last sampling date. We also looked at percent shell coverage, so not just whether that was absent or present, but we measured how much. And so in the blue line, it's average coverage. So if we look at all the turtles that we sampled, how much of their shell was covered with algae. And then if we looked at what the maximum coverage was at any sampling date, we can get an estimate that also shows this peak in July. And can we relate that to what we see with the turtles? So again, on the left-hand side, We've got a turtle that we capture in July. It's got a high percentage of algae on the shell. And then in that moment, or maybe at the next sampling date, we can see that those scutes are ready to peel off. And in this case, this is a turtle that was in the process of shedding. So as you handle it, the scutes are coming loose. And so a clean shell is right underneath. So that data that we see, which the drop off after July, is related to the physical process of the turtles growing and losing that shell scute and the attached algae. So if we compare the concentration of nitrate over time versus maximum algae coverage on the shells, we see a relationship that says that as nitrate concentration increases and peaks, we see a peak in shell coverage. But we do see a decline in nitrate concentration that it's a spurious relationship with the decline because the algae isn't declining on the scutes are declining on the turtles. They're falling off as the turtle grows. And so that percent coverage that we see into the fall is a lot to do with the turtles growing and shedding their algiferous scutes. So to conclude, we've observed a similar pattern of algae development in 3 seasons, and that pattern also matches what Whit Gibbons saw in Michigan. We find algae is very low or absent in the early spring, and it increases to a maximum in July. And that's related to lots of things, day length, it's related to nitrate concentration, it's related to water temperature. So there's lots of correlates to this, but then it declines. So that algae coverage starts to drop off after July and into August. I think someone alluded to the, probably in the alligator snapping turtle talk, they mentioned how turtles act as engineers in the environment. And so I built this slide to show the turtle's role in the nitrogen cycle. And so nitrate in the water is being taken up by algae on the turtle shell. And that algae falls off and decomposes, it becomes organic nitrogen. And so it's also important to consider the turtle as an ecosystem and as an engineer of ecosystem processes. In this case, it has a role in nitrogen management in the environment. Another conclusion slide. We find that algae is related to the nutrient status of the environment. We find it higher in the eutrophic upper pond, and algae coverage is lower in the oligotrophic lower pond. But we don't have a really independent analysis there. To say something with any kind of strength here, we'll have to sample more ponds at distance, increase our sample size to be able to say more about that. But we do see a relationship. And that indicates that there's a clear benefit to the algae, that being on the back of a turtle that is in a high nutrient environment provides a growing space for them to thrive in. If there's a benefit to the painted turtle, it's only in the time when the painted turtle has algae on its shell. To the extent that it can be camouflaged, it's only if the turtle is growing algae on its shell. If it could be a food source, it has to be on a turtle shell or another, on their neighbor's shell. So the best that we can describe for painted turtles is that this relationship is a commensalism. It's a benefit to the algae, but it's a temporary one because the scutes are shed. And that is a textbook definition of what's called a foracy. It's a type of commensalism where one species benefits and the other doesn't receive much of a benefit for that. And so I want to say at this point that it's not something that is universal to all turtles, right? There's this relationship that we're describing is specific to painted turtles, specific to painted turtles at our location, and that the nature of that relationship can vary by species and by location. And so earlier, I talked to Ethan about his alligator snapping turtles in Illinois, and he said that they have very little algae. And you can see some on the shell, but alligator snapping turtles are also referred to in other parts of their distribution as mossback turtles because of the amount of algae that occurs on there. And so there's going to be geographic variation that could be nutrient and light related, temperature related, and that the nature of the relationship is going to vary also geographically and could vary with life history. So at that point, I'll conclude and I'll take any questions that show up in the chats. Thank you for your attention.
We don't have any questions yet, but we are going to wait in just a minute. And we want to thank you, Patrick, for your presentation, and everyone else who joined us today, both presenting and watching via the live stream. And next week is the final day of the symposium. And be sure to tune in at the same time on September 24th. And our sessions are going to be human impacts on landscapes and turtles and wildlife laws, legends, and trafficking initiatives. And be sure to check out our poster session, session 2, on our website, turtlesurvival.org. And also next week at the end of the presentation will be the TCF Fund and Baylor Awards. And if we have no questions coming in yet, I think we will go ahead and say bye to Patrick. Wait, we've got one. Does time spent basking, time out of the water, influence algae coverage? And would smaller size classes have more algae due to less access to prime basking? And related, would algae coverage affect the temperature absorption in basking?
Good questions. And I'll just say that all of them are things that should be followed up on. What we do know about Basicladia, or actually it's now called Arnoldiella, the genus of filamentous algae that is on the backs of North American painted turtles, is that it is very tolerant of desiccation. It is a native algae species as far as we know. The earliest records and descriptions of algae on painted turtles and other species indicate that it's a native species. So the Basicladia or Arnoldiella, however you want to refer to that genus of filamentous algae, is very tolerant of desiccation. And that's one of the things that makes it a good colonizer of the turtle carapace. The other anatomical feature is that they have these holdfasts that actually in that first slide, you can probably see a little bit. The feet of these filamentous algae can pry into the seams of the scute and attach themselves. So they're very good at this. And unlike some other algae species, they are tolerant of desiccation. So they're on there as they bask, they can dry out. And when the turtle goes back in, they rehydrate. So they're good at that. We don't see evidence that smaller size classes have more or less algae. They certainly, if you want to compare across species, we see musk turtles or stingpots have a lot of algae relative to painted turtles. Snapping turtles have a lot of algae relative to painted turtles. But in juveniles, like especially juvenile snapping turtles, their shell texture is different and they have less algae at a young age of a similar size than a hatchling or juvenile painted turtle. So there's a lot of things that lead to variation, and it's not basking necessarily. Whether or not that has much of an impact on temperature while basking it's a good question. You would think that there's going to be some modulation of insulation due to the algae, but certainly a lot of basking turtles have denser algae mats on their back. So if you look at cooters and sliders in southeastern United States, they can be very highly covered in algae. So it doesn't seem to affect them in terms of thermal regulation. Question 4, do we know why algae turtle doesn't move to the lower pond and the non-algae turtle doesn't venture out to the upper pond? They're free to migrate in between those 2 ponds. We sample multiple times and we're not detecting individuals from one side over the other. We always assess the algae concentration and identify it to the pond. And there's no physical barrier. There's a road there and the turtles from both sides will come up and nest on that road, but they seem to have established home ranges that are on either side of it. So there's no reason other than they like where they are that we know of. Okay, thanks for your attention. And then thanks again to the TSA and all of the sponsors who made this possible. It's in a year where there's been a lot of things that have shut down. It's been great to be able to attend a conference, to participate and exchange scientific ideas. Did high school students incorporate participation in their studies? It was part of their AP class. So they get out of school, they walk up to the pond. The traps have been set the night before we pull them out, and they're learning about the concepts of eutrophication and other things as part of this study. Yeah. You're welcome. Thanks, everybody.
Thank you, Patrick. And again, thank you to all of our presenters and everyone who joined us today viewing the live stream. Be sure to join us next week at 11 a.m. daylight time. And you'll see the last day of the symposium for 2020. Thank you very much, everyone. And we'll see.
Thanks, Dave.
Thanks, Patrick.